பிரியங்கா இப்ப சாருக்கு யாரு
அப்பா சவுண்டே கேட்க மாட்டேங்குது.
Sir, very good morning. Audio will not clear, sir.
வந்துட்டாங்க சார்.
to brief about the conference.
தர்மபுரம் was established. தர்மபுரம் Good morning, all of you. Almighty Lord Nataraja, Sivagami Ammal, and Jesus. Dr. T. Aravali sir, principal of this college, simple and humble personality. Dr. K. Bonni madam, head of the Department of English. We received all the guidance from madam. Head of the Department of Zoology, Dr. V. Vanida madam, supporting in all the ways, all the faculty members of Zoology Department. Head of the Department of Physics, Dr. T. Benny Anburaj, very nice personality. All the faculty members of Physics, all the head of the Department of Dharmapuram Jnanambi Government Arts College for Women, all the faculty members of the Dharmapuram Jnanambi Government Arts College for Women, supporting staffs, all the students of this college. Last but not least, delegates and invitees, very warm good morning to each and every one of you. Dharmapuram Jnanambi Government Arts College was established by a committee of philanthropists headed by Rangavarsham IAS and Mr. T.V. Antony IAS in July 1967. Incepted with pre-university course in municipal school building, the institution evolved into one of the government women college in Nagapattinam district in those days. Now it is in MAYILADUTHURAI district. It progressed by introducing UG and PG courses subsequently towards the objectives of higher education is service to the socio-economically backward section of the society to ensure the quality, the defining elements of higher education. Our college has been periodically by the National Assessment and Accreditation Council in the third cycle. Re-accreditation of our college was awarded A grade in the academic year 2015-2016. The college marches towards the mission of empowering women through higher education with 10 UG courses and PG courses in Shift 1 and 6 UG courses in Shift 2 and 3 departments are engaged with the research. Vision women empowerment through higher education, mission, knowledge for the professional competence, humility for social commitment, truth to be the source of life, to strengthen the youth through sincere academic pursuit, and to cater to the needs of socio-economically backward women of the locality. Objectives: to provide an encouraging environment of academic excellence, to develop human potential, to promote team spirit, social responsibility, and value formation, to increase student participation in different activities, to enhance their knowledge, skills, and personality development, to generate guiding ambience towards women empowerment by which they became catalysts in all the developmental process within and outside the college campus to achieve the status of college of excellence. A research forum was established in Dharmapuram Jnanambika Government Arts College for Women, Mayiladuthurai in the year 2019. The research forum was established to create awareness of research and offer assistance to the students, academicians, researchers, entrepreneurs, and innovators to identify, protect, and manage research effectively. To bring research in among the faculty members, research scholar, and postgraduate students. The purpose of research is enhancing the society, advancing knowledge through development of scientific theories, concepts, and ideas. Aim and objectives in lines with our vision. The aim of research forum is to produce high quality, innovative, and cooperative research findings that will enrich practice and inform policy locally, nationally, and globally. To confidence to publish high quality of international renowned journals and books, to engage the dissemination and communication with the stakeholder in multiple way that maximum impact and knowledge exchange. The intellectual property right was established in Dharmapuram Jnanambika Government Arts College for Women, Mayiladuthurai in the year 2021. The intellectual property rights cell was established to create awareness and offer assistance to the students, academicians, researchers, entrepreneurs, innovators to identify, protect, and manage IPR effectively. The center actively involves in educating the importance and relevance of IPR cell for communication. The IPR cell is functioning as a nodal center for industrial entrepreneurship, education, and research for IPR cell related matters. The IPR cell is focused to conduct a certificate course on intellectual property rights and arrange a special lecture by experts in the field of IPR cell. The Dharmapuram Jnanambika Government Arts College, Mayiladuthurai, affiliated to Annamalai University, PG and Research Department of Physics, PG Department of Zoology, in association with Tamil Nadu State Council for Higher Education, Chennai, the first international conference funded by TANSCHE, virtual international conference on recent innovations on biomaterials in the current scenario. The invited speaker, Professor Rama Rao, Dr. Krishna Chaitanya, Dr. Venkateshwara, Dr. Bharati Mohan, and Mr. Madan Raj is the examiner of patents and design intellectual property of Office, Government of India. We received 95 abstracts from various institutions and more than 400 registration from Annamalai University, TPML College, ADM College, Nagapatnam, Sri Venkateshwara College of Engineering and Technology, Pondicherry, St. Joseph College of Arts and Science, Kadalur, Bharatidasan University, Sri Balaji Medical College and Hospital, Amit Institute of Pharmacy, Madhya Pradesh, Anne Velangani Arts and Science College, Government Siddha Medical College, Palayamkottai, Indian Arts Science College, Kondam, Army Ranna Government College in Namakkal, Government Arts College for Men, Nandanam, Chennai, MDT Hindu College, Tirunelveli, Madras University, Tiruvike Government Arts College, Tiruvarur, AM Jain College of Meenapakkam, Alagappa Government Arts College, Karaikudi, Government Arts Adi Kesava University, Andhra Pradesh, RGC College Tanjavur, HHD Raja College Pudukkottai, University of Kashmir, GITM University Visakhapattinam, Thiru Kolanjiyapar Government Arts College Virudachalam, Indira Gandhi College of Arts and Science, AVC College, Kalaimagal College of Arts and Science, Sambanar Koil, Idaya College from Kumbakonam. We received lot of abstract and participants throughout India. In these few words, we invite all of you and good morning to each and every one of you. Thank you very much. Thank you.
Thank you, sir. Now I invite Dr. D. Benny Anburaj, Head, Department of Physics, to welcome the gathering. Benny, sir.
Good morning to all. அனைவருக்கும் வணக்கம். Today is the day, that is உலக சுகாதார நாள், World Health Day. உலக நலவாழ்வு நாள் என்பது உலக சுகாதார அமைப்பின் அனுசரணையுடன் ஒவ்வொரு ஆண்டும் ஏப்ரல் 7ஆம் தேதி கொண்டாடப்படுகிறது. 1948ஆம் ஆண்டு இடம்பெற்ற உலக நலவாழ்வு மன்றத்தின் கூட்டம் ஒன்றில் ஒவ்வொரு ஆண்டும் 1950 இருந்து உலக நலவாழ்வு நாளாக கொண்டாடப்படுவதற்கு தீர்மானிக்கப்பட்டது. அன்றிலிருந்து உலக நலவாழ்வு நிறுவனத்தால் முக்கியமான நலவாழ்வு தொடர்பான கருப்பொருளை மையமாக கொண்டு கொண்டாடப்படுகிறது. I am very happy to announce this one. இன்று நம்முடைய கல்லூரியில் பன்னாட்டு கருத்தரங்கம் நடைபெறுவதில் இயற்பியல் துறை சார்பாகவும் விலங்கியல் துறை சார்பாகவும் உங்களை வரவேற்பால் மகிழ்ச்சி அடைகின்றேன். DG Government Arts College, Women's MAYILADUTHURAI, PG and Research Department of Physics and Department of Zoology, in association with the Tamil Nadu State Council for Higher Education, is organizing the international conference on the recent scenario, recent innovation on biomaterials, the recent scenario 2020. So I'm very privileged to stand in front of you to say a welcome address. I welcome our Honorable Principal Sir, Dr. Aravali, who made this conference in a very successful manner. I'm very much thankful to you, sir. I welcome you, sir. And our research advisor, Dr. Ponni Madam, Head of the Department of English, I also welcome on behalf of this organizing committee members. I welcome you, Madam. And Head of the departments of all the college and neighboring college department and participants and the organizing committee, well managed, they arranged the manner. I'm very much impressed and thankful to the organizing committee once again. And I welcoming the committee, that is resource persons, Ramarao Mulla, Kamalagaran Rao, and Dr. Krishna Saitanya, Dr. K. Venkateswara Rao, Dr. Bharati Mohan, and Dr. Madan Raj, and Network Administration Bala, Global Infotech, Kumbhakonam, and the participants, students from research as well as the PG students of our own college and neighboring institution, not only state, all over the world, internationally, they participate. I very much impressed. Welcome one and all, everyone. I mean, thank you very much for the organizers, Dr. Aidamala Selvi and Dr. Muthulingam, for well managed after the quarantine period. It's a very nice pattern, that is the topic. It's a need to this world, because only one sentence I would like to say. Are we able to reimagine a world where clean air, water and food are available to all? Just think பண்ணி பாருங்க. 50 years முன்னாடி உள்ள உலகம் இப்பொழுது இருக்கிறது. நம்ம சுத்தமான நீர் கிடையாது, சுற்ற காற்று கிடையாது. இருந்தாலும் as a scientist we are taking the very good steps to achieve all these things. I wish the organizing committee and committee members and especially our principal sir and research advisor, I welcome you one and all. Thank you very much for this opportunity.
Thank you for your warm welcome. Now I invite our most respected principal sir, Dr. D. Aravali, to deliver the presidential address.
Morning to all. Respected research advisor, head of the Department of English, Dr. Ponni Madam. Respected head of the department. Respected head of the Department of Physics, Dr. D. Benny Anburaj sir. Respected head of the Department of Zoology, Dr. Vanida Madam. Respected International Conference Organizing Secretaries, Dr. M. Muthulingam, Associate Professor of Zoology, Dr. R. Aida Malachelvi, Assistant Professor of Physics, all the head of the Department of Dharmapuram Yanambiya Government Arts College, all the faculty members of our college, most respected guest speakers today, Professor Ramara Malla, Professor Dr. K. Kamalagara Rao, Professor Dr. Krishna Jantania, dear participants of the international conference and the students, a very good morning to all of you. It is a great pleasure to welcome all the participants today for the 2 days international conference. I welcome all the eminent speakers and guests from all over the country, from different walks of life. You have come here to share their knowledge and vast experience with the students community. This is the first international conference in the history of our college, which is completely conducted on a digital platform. We are proud to announce that this international conference is being conducted in association with the Tamil Nadu State Council for Higher Education. At the outset, it's my primary obligation to express my sincere thanks to all the renowned professors from Department of Physics and Department of Zoology who have got involved in the remarkable event, which is going to bring us a greater amount of reputation and famous. At a government arts college, it is not an easy task to conduct international level seminar conference in a successful way. Why? Because we have lack of infrastructure facilities. Apart from all the hurdles, Professor Dr. M. Muthulingam from the Department of Zoology, Professor Dr. R. Aida Malachelvi from the Department of Physics, who have taken maximum amount of endeavors so as to conduct this marvelous event in our institution. Being the head of this college, I feel elated to be the part and parcel of the wonderful conference. This is an excellent event to bring together leading academic scientists and research scholars to exchange and share their experiences and current scenario research outcomes to all the aspects of biomaterial. This conference also offers innovations, trend, and concern apart from the practical challenges encountered and solutions adapted in the advancement of biomaterials. Among all other science subjects, physics is known as the king of science. So this is the right platform to learn a lot. You all have the bright opportunity to become masters in physics. Please make use of the opportunity extended to you. Before conclude my address, I would like to thank all the staff members, head of the Department of Physics, and all the staff members and head of the Department of Zoology, and Organizing Secretary, Dr. M. Muthulingam and Dr. Aida Malarchelvi. I wish this event a grand success. Once again, I extend my sincere thanks to one and all. Thank you very much.
Thank you, sir. Thank you for your enlightened message. Thank you for your support and encouragement to make us to do a Himalayan victory. Thank you very much, sir. Now I invite our respected Madam Research Advisor, Madam Dr. K. Ponni, to deliver the special address. YouTube கொஞ்சம் ஸ்லோவா தான் அதனால
Good morning and greetings to all around me. Dharmapuram Jnanambike Government Post College for Women is a multidisciplinary higher education institution situated on the banks of River Cauvery, which has its own heritage. Our college serves the women of almost 11 educational districts in and around Mayiladuthurai, with the vision of empowering women through higher education. Today, we set a milestone by conducting this international conference with the patronage of our principal, Dr. D. Aravali, and the organizers, Dr. M. Muthulingam, Associate Professor of Zoology, and Dr. R. Aida Malarchelvi, Assistant Professor of Physics. Hearty welcome to the eminent resource persons and efficient research scholars, our teaching fraternity, members of the research forum, and my ever dear students. The title of this virtual conference is Recent Innovation on Biomaterials in the Current Scenario. தற்கால கண்டுபிடிப்பில் உயிர் பொருட்களின் நவீன நிலைப்பாடு. It is organized jointly with Tamil Nadu State Council for Higher Education. It has its own significance in integrating science and society. At this juncture, I remember Bharat Ratna APJ our former president and premier scientist who wanted to upgrade the quality of life with the application of science. He created centers of nanotechnology and advanced biomaterials to initiate this process. The technological vision of Abdul Kalam has proved its success in the technical functionality of biomaterials found in the implant substances of contact lenses, pacemakers, heart valves, orthopedic services and devices, and so on, like organ and tissue repair. RICBCS 2022 would be an excellent interdisciplinary platform to to bring together leading academic scientists and research scholars to exchange and share their experiences and current research outcomes to all aspects of biomaterials. It is a great pleasure for me to welcome our speakers today, Professor Rama Rao Malla, Department of Biochemistry and Bioinformatics, Geetham University, Visakhapatnam, Andhra Pradesh, Dr. K. Krishna Chaitanya, Associate Professor in Chemistry, Aksum University, Aksum, Ethiopia, Dr. K. Kamalakara Rao, Department of Biochemistry, Adikavi Nanaya University, Rajamundry, Andhra Pradesh. Dr. K. Venkateshwara Rao, Professor of Nanotechnology, JNTUH Center for Nanoscience and Technology, Institute of Science and Technology, Professor of Nanotechnology and Controller of Examination, CNST, Institute of Science and Technology, Kukatpalli, Hyderabad, and Dr. D. Bharati Mohan, Associate Professor, Department of Physics, Pondicherry University, Pondicherry, and Mr. M. Madan Raj, Examiner of Patents and Designs, Intellectual Property Office, Government of India. The untiring efforts of the organizers is applaudable. Let this conference to be an eye-opener to the budding researchers and a bridge to the scientists to enable sufficient stability in the practical applications. Wishing each and everyone a good day. Thank you.
Thank you, ma'am, for your motivating speech. Now I invite our organizing secretary, Dr. M. Muthilingam, for the introductory note of the today's speaker, Dr. K. Krishna Chaitanya. Thank you. Is he entered? Is he entered?
Krishna.
Yeah.
Once again, good morning to every one of you. Our invited speaker, Krishna Chaitanya, Associate Professor, Department of Chemistry, College of Natural and Computational Science, Aksum University, Ethiopia. He is working as an associate professor and rich experience with the teaching, 15 years of teaching experience, actively involved in conducting various research and community service-based funding projects in the field of biochemistry, chemistry, nanotechnology, medicinal chemistry, natural product, molecular biology, and infection biology. The trained base advanced and translational molecular biochemistry, synthetic chemistry, medicinal chemistry, material chemistry, and immunology courses by world-renowned chemists, immunologists, and biochemists such as Professor Abul Abbas, Professor G.P. Talwar, father of Indian immunology and biochemistry, and other esteemed Nobel Prize winners and international and national faculties. Actively participated in training for the writing grants and research proposal in advanced and translational research by making national and international collaboration as well as submitting the manuscript to be reviewed research channel. And the research specialization is mainly immune tolerance, anti-cancer, anti-inflammatory agents, nanoparticles, and antimicrobial synthetic agents, as well as the therapeutic molecules from the medicinal plants, the controlling and treatment of cancer, auto-inflammatory related and infectious disease, understanding biomolecular interactions and improve the nanomaterials for target drug delivery in female reproductive and breast cancer by targeting tumor microenvironment molecules. He has completed many projects and collaboration with Tamil Nadu Agriculture University, Andhra University, Central University of Kerala, and he attended many training courses. The hands-on training like RT-PCR, real-time PCR, plotting techniques, FACS analysis, FTIR, NMR, TLC, chromatography, etc., etc. And he attended and conducted many workshop courses. The conference and seminar he attended more than 20 conference he is attended and published more than 75 reputed national and international journals is published. With these few words, I welcome Dr. Krishna Chaitanya from Ethiopia to deliver invited lecture from the gathering. Thank you very much. Now I invite Krishna Chaitanya for the wonderful event. Thank you, thank you. Krishna, Professor Krishna. Professor Krishna. Professor Krishna. இல்ல இப்ப இப்ப கிருஷ்ணா
Let us welcome Dr. K. Krishna Chaitanya for this conference. Thank you, sir. Welcome you, sir. Sir, kindly unmute your phone, sir. Kindly unmute.
Can you audible now?
Yeah,
Hello?
sure, sir. Yeah, hello.
Yes, yes, madam. Can I start my presentation?
Please, sir.
Good morning. So good morning, everyone. Yeah, myself, I'm Dr. Krishna Chaitanya, and now I'm taking presentation on dual inhibitors, development of the dual inhibitor. And everyone audible? It is audible? Hello?
Audible, sir. Proceed.
Yeah. Myself, I'm Dr. Krishna Chaitanya. Now I'm sharing the slide, madam. Slide sharing అలాగా slide share అలాగా Google Meet
sharing. Meeting sir.
అవును సార్
Hello.
Google
Share option. Share address.
సాధారణ సార్ Sir, wait, sir. I'm sharing the slide. Let's do some slide share outlook. Slide share slide Google Meet. ఇప్పుడు నాకు పీపీటీ ఇక్కడ ఒక మన షేర్ Sir, it is okay? Hello?
No, sir. Go ahead.
Yeah.
Yeah.
Sorry for disturbance, madam. My name is Dr. Krishna Chaitanya. I'm working as associate professor in University of Oxford University. Yeah, sorry for delay, sir. So due to some problem. Yeah, my title is, and first of all, I'm congratulating to all the participants as well as organizers. And now I'm starting my presentation. My title is In Vitro Screening of potato dual COX-1 phylox inhibitors from selected medicinal plant as a novel anti-inflammatory agent. First of all, I am going to introduction, statement of the problem, objective, significance of the study, methodology, results, as well as discussion. First of all, you know very well, inflammation is a part of innate immunity. In inflammation is a protective. In inflammation, it is protective against both infectious as well as non-infectious diseases. But during inflammatory, what is the meaning of inflammation? Means inflammation is a type of innate immunity in which we have to protect against both infectious as well as non-infectious organism. Important inflammation having two different parts. One is acute inflammation, another one is chronic inflammation. During acute inflammation, it is a protective response, but during chronic inflammation, it is causing so many disorders, chronic inflammatory related disorders like cancer as well as diabetes. During inflammatory response, the inflammatory mediators such as interleukins, you know very well, interleukin B, interleukin 6, interleukin 8, as well as so many necrosis factor and nitric oxide are responsible for causing different type of the diseases. We are having two different type of inflammation. During acute inflammation, these inflammatory mediators levels are normal or basic. During chronic inflammation, the levels of chronic inflammatory mediators are increasing. Okay, these chronic inflammatory mediators such as cytokines as well as nitric oxide, which also responsible for upregulation of cyclooxygenases as well as phyloxes. Those enzymes are responsible for synthesis of prostaglandin as well as leukotrienes. These leukotrienes as well as prostaglandins responsible for upregulation of inflammatory diseases as well as the different turmeric factor. You know, here very important factor what I am telling. Okay, inflammation is protective, but when acute inflammation became chronic inflammation, it is causing different type of the diseases due to upregulation of unwanted inflammatory cytokines like interleukin 1, interleukin 6, and TNF alpha, interleukin 8 as well as nitric oxide. And these inflammatory cytokines also responsible for progression of different type of the cancer. These chronic inflammation is due to merely imbalance of both pro-inflammatory cytokines as well as anti-inflammatory cytokine. Here, when upregulation of pro-inflammatory cytokines like interleukin 1, interleukin 6, interleukin 8 as well as TNF alpha is responsible for downregulation of anti-inflammatory cytokines like TGF beta as well as interleukin 10. That's why imbalance between inflammatory mediators as well as anti-inflammatory mediators responsible for causing different type of the diseases. Here my important focusing on when pro-inflammatory mediators are upregulated, it activates other inflammatory enzymes like cyclooxygenase 2 and lipoxygenases. These COX as well as lipoxygenase responsible for synthesis of the prostaglandin as well as the leukotrienes. These prostaglandins and leukotrienes are responsible for causing inflammatory diseases. Here, very important point is, you see, when acute inflammation became chronic inflammation, these chronic inflammatory mediators such as pro-inflammatory cytokine responsible for upregulation of COX as well as the LOX. These COX as well as the LOX, which can prevent the progression of this, which can make more cell cycle. It activates the cell cycle all pathways, like it activates unwanted activation of cell cycle by progression of checkpoints, as checkpoints, G1 checkpoint, G2 checkpoint, and that's why what is my meaning, when more inflammation is there, these inflammation responsible for synthesis of the prostaglandins as well as the leukotrienes. These leukotrienes and prostaglandins activate unwanted cell signaling pathways. These unwanted cell signaling pathways responsible for activation of S pathway. You know, S pathway means synthetic pathway responsible for DNA synthesis. The uncontrolled checkpoints, uncontrolled activation of this S phase is responsible for cell cancer, and it also inhibits the apoptotic pathway, and it also responsible for resistant of drugs, multiple drug resistant against cancer cells, and stimulate the angiogenesis, and also responsible for activation of vascular endothelial growth factor. These growth factors are responsible for the migration of tumor from one place to another place, and it's also responsible for the conversion of benign tumors are converted into malignant tumors. And very important thing here, my important topic is there is a relation between inflammation as well as cancer. If there is the inflammation as well as the cancer, if there is the acute inflammation is a protective, when acute inflammation more than 3 to 5 days, it becomes chronic inflammation. During chronic inflammation, it releases different inflammatory mediators. These inflammatory mediators upregulate the inflammatory enzyme. These inflammatory mediators instead of also upregulate other mechanisms responsible for progression of the cancer. Here, my important thing is by targeting inflammation at the initial level. By targeting inflammation at the initial level, it is very useful to control the cancer progression at the next level. That's why the scientists for controlling inflammation, they're using both steroidal as well as the non-steroidal anti-inflammatory drugs. These drugs are responsible for treatment of the diseases, but these drugs are having both beneficial effect as well as the harmful effect. When you are go for the glucocorticoids are the hormone which are responsible for decreasing the inflammation by down regulation of the key inflammatory mediators as well as their molecular pathway, responsible molecular pathway like nuclear factor kappa beta. You know very well, we are having the different cytokine like interleukin 1, interleukin 6, and TNF alpha. These cytokines, they utilizes NF kappa beta pathway. This NF kappa beta pathway instead activate inflammatory mediators, and also it also responsible for consumption of more ATP by cancer cells. And you know, another important thing, we are having the two different drugs are responsible for to stop the inflammation. One is steroidal drugs as well as the non-steroidal drugs. But steroidal drugs having some disadvantages, but it having some advantages. But initially it cures the inflammation as well as inflammation related cancer. But the major disadvantage is it is also causing some side effects. You see, when we are going we are having two different drugs are there. One is steroidal. Already we discussed about the steroidal drug like glucocorticoid, but another drugs are called non-steroidal anti-inflammatory drugs. These non-steroidal anti-inflammatory drugs are developed with a different group of the scientists due to some disadvantages in steroidal hormone side effects. We are having two different type of the steroidal drugs are there. One is COX-1 related steroidal drugs as well as the COX-2 related steroidal drug. But important thing is here one of the disadvantage of non-steroidal anti-inflammatory drug due to this non-steroidal inflammatory drug, those drugs it act on both COX-1 as well as the COX-2. Before going to these disadvantages, I'm telling about the COX. We are having the three different enzymes are there. One is phyloxoxygenase, another one is COX. COX means cyclooxygenase 1 as well as cyclooxygenase 2. Here, very important thing is you see non-steroidal anti-inflammatory drugs which act on both COX-1 and COX-2 due to its similar structure, because COX-1, COX-2 are isoenzyme. Due to this isoenzymic nature, those enzymes which share active site. Due to sharing of these active site, non-steroidal anti-inflammatory drugs it act on both COX-1 as well as the COX-2. That's why it is having disadvantages, because COX-1 is responsible for to maintain normal intestine function, but COX-2 is inducible cyclooxygenase, which is responsible for upregulation of the cancer. That's why now the major disadvantage here, when we see non-steroidal anti-inflammatory drugs, these non-steroidal anti-inflammatory drugs it act on both COX-1 as well as COX-2, but COX-1 is a protective enzyme which present in our body to prevent the unwanted to prevent the gastric irritation. But COX-2 is inducible cyclooxygenase. COX-2 is called inducible cyclooxygenase, which activated by the pro-inflammatory cytokine responsible for progression of inflammation as well as cancer. But here these non-steroidal anti-inflammatory drugs which act on both COX-1 as well as COX-2, that's why it having disadvantages of to causing different gastrointestinal complications such as mucosal damage and bleeding. It is related to COX-1 because COX-1 is responsible for maintenance gastrointestinal normality, but due to blockage of COX-1, it unable to perform its function. It will be blocked by the non-steroidal anti-inflammatory drug. That's why the scientists, due to this disadvantage of non-steroidal anti-inflammatory drugs, the scientists they want to develop the drug by targeting only COX-2. Okay, you know the different scientists they developed the different selective COX-2 inhibitor. These selective COX-2 inhibitor which are responsible for only only targeting COX-2 without having any effect on the COX-1. That's why these drugs are succeeded. Different clinical trials also reported that different clinical trials also reported that it significantly decreases different cancers like colorectal cancer as well as the breast cancer and the lung cancer also. That's why the important thing is we are having, when you are coming, you see this is my statement of the problem. When you are seeing this pathway, you see arachidonic acid, the fat, unsaturated fatty acid. This fatty acid is a precursor for these inflammatory mediators like leukotrienes as well as thromboxins and leukotrienes as well as the thromboxins as well as inflammatory mediator. Okay. Here, my important thing, what is my target is, you see, the non-steroidal anti-inflammatory drug which act on both phylox as well as COX-1 as well as the COX-2. But when it acts on the COX-1 as well as the COX-2, this COX-1 is responsible for maintain normal functioning of gastrointestine. When the dual inhibitor which act on the COX-1 as well as the COX-2, it is having some disadvantages. That's why the scientists they prefer to develop the COX-2 inhibitor. That's why you see we are having some disadvantage of non-steroidal anti-inflammatory drugs. That's why the scientists are forced to develop the selective COX-2 inhibitor. These selective COX-2 inhibitors are responsible to prevent the COX-2 related inflammatory mediator and COX-2 related cancers. And you see another important thing. This latter, the different scientists they once again they worked on the COX-2 selective COX-2 inhibitor, and they find out different disadvantages of these COX-2 inhibitors also, because it is not sufficient to safe use for cancer treatment as a chemopreventive therapy, because these COX-2 inhibitors having the major disadvantage by upregulation of phylox. That's why the scientists reported that both non-steroidal anti-inflammatory drugs as well as the selective COX-2 inhibitors they prevent the prostaglandin without modulating phylox enzyme. That's why when phylox enzyme is when it only affect on the COX-1 as well as the COX-2, but it does not have any effect on the phylox. That's why due to this problem, the phylox enzyme is activated and all arachidonic acid pathways shift towards the phylox, and which is responsible for the synthesis of leukotrienes as well as the thromboxin. Leukotrienes. These leukotrienes are responsible for causing different cancer. Here important thing is my statement of the problem. We are having the different inhibitors are there. First one is non-steroidal anti-inflammatory drugs as well as the steroidal anti-inflammatory drugs. These non-steroidal and steroidal anti-inflammatory drugs which can decrease inflammation by inhibition phylox as well as the COX-1 as well as the COX-2. The scientists initially they focused on the phylox, but when phylox is inactivated, but these carcinoid acid pathway it shift towards the COX-1 as well as the COX-2. When the scientists developed the COX-2 inhibitor, it shift towards the phylox as well as the COX-2. That's why the scientists they want to develop the drugs which affect with inhibitory effect on phylox as well as the COX-2 without having any effect on the COX-1. My important thing is, you see, I want to develop the phylox and COX-2 inhibitor. We are having, you see, here important when you are seeing this slide. Initially the scientists they developed the classical non-steroidal anti-inflammatory drugs. These classical non-steroidal anti-inflammatory drugs it act on both phylox, COX-1 as well as the COX-2. But it is having advantage, but disadvantage is here intestinal problems are coming. That's why intestinal bleeding is coming. That's why the scientists they want to develop the drug without having any effect on COX-1. That's why the scientists initially forced to develop the drug against phylox, and after that the scientists also forced to develop the drugs on phylox. But here the initial scientists when they developed the drug against only phylox, the phylox enzymes are inhibited, and all arachidonic acid pathway shift towards the COX-2, and immediately COX-2 enzymes are activated. That's why the scientists they developed the phylox inhibitors as well as COX-2 selective inhibitors. That's after that the scientists that developed the COX-2 selective inhibitors. These COX-2 selective inhibitors also having some disadvantages because it shifted all the pathways towards the arachidonic acid pathway. But here important thing is our statement of the problem is our main statement of the problem. We have to develop the different therapeutic drug without affecting COX-1, by affecting both 5-LOX as well as the COX-2. That's what it's called dual inhibitor, novel dual inhibitor. That's why you see when you are seeing this pathway, this is arachidonic acid pathway. Once during the stimulators, during cancer condition or during inflammatory condition, here PLA2 is activated. PLA2 means phospholipase A2. This phospholipase A2 acts on the phospholipids. It releases arachidonic acid. This arachidonic acid is a substrate for two pathways. One is 5-LOX pathway, another one is COX pathway. Here we are having the two different COX are there, COX1 as well as the COX2. Here our inhibitors should not affect on the COX1, because when it affect on the COX1, we are getting different problems. That's why the scientists are importantly developed the drugs by targeting COX2 and by targeting 5-LOX. Here different lipoxygenase also there, 15 lipoxygenase, 5-lipoxygenase as well as 12 lipoxygenase are there. Among all lipoxygenase are responsible for releasing leukotrienes. These leukotrienes L4 as well as the B4 responsible for upregulation of cancer. As well as you see, when you see this pathway also, this COX-2 also releases prostaglandin, different prostaglandins as well as the thromboxane. These molecules specifically react with bind with the receptor, and it activates the different cancer pathways and upregulate the cancer. You see here, plus means pro-tumorogenic, minus means what? Anti-tumorogenic. Okay, you see COX-1 is anti-tumorogenic, COX-2 is tumorogenic. That's why we have to target COX-2 as well as 5-LOX for development of the novel drug. You see, important here, very important thing is, you see, when our drugs are developed, when our drugs are targeted COX-2, okay, what happened? COX-2 mediators are inhibited. COX-2 related prostaglandins are downregulated. When COX-2 related prostaglandins are downregulated, automatically 5-LOX enzymes are upregulated. Here, major important problem is once we target the COX-2 is inhibited. Once COX-2 is inhibited, this more amount of the substrate which is responsible for production of leukotrienes by react with the different 5-LOX. That's why here once again you see this point. You see, say example, once COX-2 selective inhibitors are responsible for activation of the 5-LOX. This 5-LOX is responsible for synthesis of the leukotrienes and cysteine leukotrienes. These leukotrienes and cysteine leukotrienes are responsible for causing more inflammation as well as causing proliferation by inhibiting what? Apoptosis. You see, say example, once we targeted COX-2 related prostaglandins are decreased, but what happened here? Leukotrienes are increased. Cysteine leukotrienes are increased. When cysteine leukotrienes are increased, automatically it is responsible for causing or upregulation of what? Cancer. That's why the scientists they want to focus on the development of the drugs which inhibit both COX-2 as well as 5-LOX. That's why my important objective is I want to develop the drugs which act on the COX-2 as well as 5-LOX as anti-inflammatory mediators. You see, need for development of novel plant-derived COX-1 as well as COX-2. What is the need? You see, here important need is, you see, drugs which act on both COX-1 as well as the 5-LOX. But here important, some drugs only act on the 5-LOX. They are unable to act on the COX-2. Okay, this is one we are finding the different disadvantages. Some drugs only affect on the COX-1, 5-LOX. Once it affects on the 5-LOX, it leads to upregulation of the COX-2, and this COX-2 responsible for increasing pro-inflammatory mediators. When we target the COX-2, same problem. That's why we are better to target both COX-2 as well as the 5-LOX to prevention of inflammation and to prevention of or progression of the cancer. That's why because I am working on the medicinal plant. Traditionally different medicinal plants are responsible for treatment of different inflammatory related diseases, like inflammation having the 4 important characters: Romer, Chober, Collar, Dollar. An important thing is here edema. Edema means very well, Here, romer means, redness, tumor, and collar as well as what? Dollar. And these abnormalities are responsible for, these abnormalities are the signs for inflammation. That's why the different people who are living in villages in Ethiopia, they 100% depend upon the traditional medicine system. They use different plants for treatment of different inflammatory related diseases, because these medicinal plants having high safety. That's why in Ethiopia, since a long period, different community people use medicinal plants for treatment of the diseases. Okay, there is a, because it is having easy to use, and it is having more accessibility and biomedical benefit. Based upon the traditional important, I selected the 10 different plant among the 10 different plant, but this Minthanes herbifolia, it is giving a better result for development of both phyllox as well as the cockstool. You see this plant related to family of Clechaceae, and these leaves are generally used for treatment of both bacterial as well as fungal pathogens responsible for skin diseases. That's why I selected this plant, because you see here very important thing is, and here infection leads to inflammation, and inflammation leads to cancer. That's why in Ethiopia, different culture people, different traditional people, they use the medicinal plant treatment of different diseases. But this Minthanes herbifolia, especially used for treatment of bacterial as well as the fungal infection. These bacterial and fungal infection responsible for upregulation of chronic inflammation. This chronic inflammation is responsible for cancer also. That's why it is traditionally it is useful for management of inflammatory diseases. And here, hence my present study, it contribute to fill the existing gap to determine the anti-inflammatory activity of the selected traditional medicinal plant Minthanes herbifolia in Aksum city. This plant is most available in entire Ethiopia, but I selected this medicinal plant leaves from Aksum city. This is our university is located in this city. I selected this plant, and therefore the present study was evaluated to inhibitory effect on Minthanes herbifolia leaf extract, organic leaf extract, by using COX-1, COX-2, phyllox enzymes, inhibitory activity as well as phytochemical analysis. Based upon the my proposal, I developed the three different objective, the general objective as well as the specific objective. And in general objective, I development of COX as well as the phyllox inhibitor from the medicinal plant. In the specific objective, I'm having two different specific objectives are there. One is screening of all extract against COX-1, COX-2, and phyllox as well as qualitative analysis. Because I selected the medicinal plant, I make into the 4 different extract. These 4 different extract I selected against COX-1, COX-2 as well as the phyllox. And after the performing of this experiment, I calculated the percentage of inhibition and IC50 values. And after that, I done for phytochemical analysis, qualitative as well as the quantitative. But I am here mentioning about qualitative, and I done quantitative also, and GCMS also. I done the GCMS analysis also. And you see, this study is helpful to fulfill the plant-derived development of plant-derived anti-inflammatory agent by targeting phyllox as well as the COX-2 mediated pathway. You see, this is the background of materials and methods. I already discussed about you. The study area is Aksum city. Aksum University. Aksum city means our university located in Aksum city. I collected plant material from Aksum city, and authentication also done by the botanist. And after the collection of the plant material, I make into the authentication. After authentication, I collected the leaf material, because leaf materials are traditionally used for the treatment of inflammatory diseases as well as the cancer. And these leaf materials I go for authentication, and primary authentication occurs in our department, Aksum University. And later I send a Central University, Addis Ababa University, for further clarification. And after identification, and after authentication of the plant material, then I take the plant. I take the plant. I go for the drying, and I make into the powder. I prepare the plant extract by using the 4 different solvent by using the Soxylate method. Okay, I used the 4 solvent here. One is petroleum ether, chloroform, ethyl acetate, or acetone, as well as ethanol. By using the 4 different solvent, I make the plant extract, and after that I measure the percentage of yield, then I go for phytochemical analysis, and after that I go for enzymatic assay kit, Phylox as well as the COX1 as well as the COX2. Here, very important thing is Phylox enzyme inhibition assay developed by the Redden et al. Because I finished my part of PhD. I done my part of PhD in University of Hyderabad under the guidance of Dr. Redden. Is a person first initially started different enzymatic assays in University of Hyderabad. Based upon his methodology, I little modified this methodology. And here important thing is, you know very well, enzyme inhibition means I have to take three things. The first one is enzyme, and the second one is inhibitor. Inhibitors means here plant extract, and another one is called substrate. You know very well, enzymes are react with the substrate. Enzymes are react with the substrate, and it gives the product. Like Phylox as well as the COX1 and the COX2. These are the three different enzymes are there. Three different enzymes react with the arachidonic acid pathway. When it is react with arachidonic acid pathway, it produces prostaglandin. Once again, I want to give a clarification. You see here, you know, it is called phospholipid. It is called phospholipid. It is the precursor, like it is the precursor. This phospholipid is the precursor for arachidonic acid. This arachidonic acid is a substrate for two different enzymes here. One is Phylox, another one is COX2. Okay. Once Phylox is interact with this substrate arachidonic acid, it produces what? Leukotrienes. And same thing, when arachidonic acid interact with the COX2, it also produces what? Prostaglandin. Here, my important thing is I want to develop the novel Phylox as well as the COX2 drugs by inhibiting these interaction. Which interaction? The interaction between arachidonic acid as well as the Phylox, as well as the interaction between COX2 as well as the arachidonic acid pathway. That's why you see here I take first of all I take the different 200 microliters of different concentration of plant extract. This plant extract I kept in a buffer, and plant extract is ready. Plant extract means medicine, and this medicine is treated with enzyme lipoxygenase, and after that we have to check the enzyme inhibitory activity based upon the nanometer. Then it is also same, you know very well. Here also same thing. Our plant extracts are treated with different enzymes like COX2, and we have to check the color reaction. And here, important thing is I'm coming to the discussion. You know very well. I selected the Menthala serpifolia. This Menthala serpifolia make into the four different extracts: petroleum ether, chloroform, acetone, as well as the ethanol. Four different extracts because I selected four different organic solvent based upon their polarity, less polarity to more polarity. Petroleum ether is low polarity and ethanol is more polarity. And I got different colors also, consistency and the percentage of yield. And based upon the result, the acetone exhibit more percentage of yield, 7.8 grams per 100 gram. Later ethanol. After that, after finishing a percentage of yield, I go for the qualitative analysis by using the different reagent, and based upon the qualitative analysis, we have very important thing. You know, phytoconsistent. We are having two different type of phytoconsistents are there. Primary phytoconsistent, primary metabolites as well as the secondary metabolite. Here I am only checking the secondary metabolite because these secondary metabolites are responsible for fighting against plant pathogens. Because these secondary metabolites are present in the plants. These secondary metabolites are responsible for to mediate immunity or to mediate protection against plant infections. That's why you know very well these are having different biological activity, anti-cancer activity, anti-inflammatory activity, antioxidant activity. That's why I done all qualitative phytochemical analysis with the 4 extract because all sample containing all the secondary metabolite, but among my test, acetone as well as ethanol containing the more amount of the secondary metabolite. Here, 1 star means presence, 2 star means moderate, as well as 3 star means presence of more amount. And after that, I went for quantitative analysis. Based upon the quantitative analysis, by plant extract containing more amount of the flavonoids as well as the tannin. You know very well, flavonoids and tannins are responsible for to mediate anti-inflammation as well as the anti-cancer agent. That's why, based upon these phytochemical analysis, we reveal that the ethanol, acetone as well as ethanol leaf extract of Menthala serpifolia containing the more amount of the secondary malats such as flavonoids as well as the tannins. You see, and after that, after finishing phytochemical analysis, and I conducted the phylox enzyme. Already I discussed about the methodology, and you see, I'm having how many extracts are there? I'm having the 3 different extracts are there. So I'm having 3 different extracts are there here. P means Minthanas. MA means Minthanas arbofolia leaf plant extract. Petroleum ether. It is MA means Minthanas arbofolia. L means leaf. Petroleum ether, chloroform, acetone as well as the ethyl estate. You see, when I test here, I'm taking different concentration 20, 40, 60, 80, and 100 per presentation purpose. I only here mentioned about 100 microgram per ml. You see, when you are seeing when I'm seeing inhibitory activity of these 4 extract compared with standard ethyl estate exhibit good percentage of inhibitory activity with a significant IC50 value, because here very important thing is acetone also exhibiting inhibitory activity. Here important thing is here I'm taking 20 to 100 microgram per ml of extract, all extracts exhibiting dose dependent inhibitory activity, but among all extract but ethanol extract exhibiting good inhibitory activity, 77% of inhibitory activity with IC50 values of 23. You know very well, we should consider about IC50 value. Here IC50 value is when it is having our therapy, or you know very well, therapeutic molecule, all therapeutic molecule, they should exhibit more percentage of inhibition with less IC50 value. Here, the therapeutic consideration, according to therapeutic consideration, here ethanol exhibiting more percentage of inhibition as well as less IC50 value. Then you see my reports also similar to previous reports of my other colleagues, my other colleague like Sastri et al. Okay, he reported that methanolic seed extract and isolated compound exhibiting good phylox inhibitory activity with less IC50 value. You see, when compared to my sample, it exhibiting good activity because he isolated different phyto compounds from the methanol and tested. Here, important thing is methanol extract also giving good result as well as the isolated compounds also giving a good result. That's why methanol is containing anti-inflammatory molecule, methanol extracts. Same thing, phylox inhibitory activity of methanol extract also. These inhibitory activity due to presence of all these compounds. That's why here important thing is my work is having strong correlation with different reports. Here I reported more significant reports. Then here COX-1 as well as the COX-2. Here, you know, very important point is here. Very important point, we are having the 2 different cyclooxygenases are there, COX-1 as well as the COX-2. That's why our plant extract we have to test against both COX-1 as well as the COX-2. But here important thing is our plant extract should not more act on the COX-1. We should act on only COX-2. That's why we have to test it against COX-1 as well as the COX-2. Initially we tested against the COX-1 and we got this result. And this result you see a petroleum ether and a chloroform as well as acetone as well as ethanol as well as the standard. Here different concentration already I discussed it before, and here all extract giving all extract exhibit dose dependent inhibitory activity. You see petroleum ether exhibiting 23% of inhibitory, 31, 44 and 51. Here very important point is here ethanol extract exhibiting good percentage of inhibition when compared to the remaining extracts. It's showing a significance also, and it is also exhibiting a less IC50 value of 93. You see, this is a diagram. This is I selected ANOVA, 2 ANOVA, a comparison between standards as well as different extracts, standards, and I comparison between petroleum ether, chloroform, acetone as well as ethanol as well as the standard. Based upon the standard, I got the significance values. And it is the COX-2. I'm also tested with the COX-2. When compared to the COX-1, the COX-2 is more inhibited. You see, already I told about you, I mentioned to you, I selected 10 different medicinal plants to check both phylox, COX-1 and the COX-2. Among all the plant extract, Minthanas arbofolia, it is giving good result against phylox as well as the COX-2 without interfering COX-1. That is major thing, because I selected remaining plant. It also act against COX-1. That's why when compared with all plant extract, the Minthanas portfolio showing a good selective COX-2 inhibitory activity and the phylox inhibitory activity. Okay, as shown in the figure 2, the 4 different extracts also showing good activity. Among all the activity, last ethanol extract exhibiting the 65% of activity with less IC50 value. Then you see, based upon the result, you see very important thing is here in vitro cyclooxygenase inhibitory activity. Here when compared, I done both COX-1 as well as the COX-2. My plant extract, because you see here important point what I'm telling, COX-1 and COX-2 are isoenzymes. COX-1 and COX-2 are isoenzymes. They share the same active site. If any plant extract which affects on the COX-2, it also affects on the COX-1. But here important thing is due to some molecular dynamics or molecular interaction. Okay, my plant compound, it more act on COX-2 when compared to the COX-1. This is more advantage. And you see my reports also, my results also, my results also are correlated with the different reports are reported by this is the Bharati et al. See, there are another group, already I told about you, Dr. Redden, University of Hyderabad. They're doing, they're also developing different novel dual inhibitor. They're working on different plant extract, and they identified different phylox as well as the COX-2 inhibitor from Terminia species. That's why this is based upon their my topic. Based upon their my topic. Hello.
Yeah, proceed.
Hello.
Hello, proceed, sir.
Yeah. That's based upon their. Wait, man.
Krishna, good afternoon, Krishna.
Yes, sir.
Thank you very much for your wonderful speech.
Yes, sir. Yes,
thank
wait,
you.
Sir. Wait. 1 minute, sir. 1 minute. Yes, some disturbance are there.
Professor Ramarao.
Sir, I am ready. If we can left this presentation, I can join.
Very sorry for the delay.
Okay, no issue, problem.
Yes, sir. Any question, sir? Any question?
Audience,
Any question?
please rectify. Please ask any question.
Hello, this is yeah.
Okay, sir. No more questions. So thank you very much.
Okay, sir. Thank you, sir.
Okay. Thank you, Professor Krishna.
Yes, sir. Thank you.
Now I welcome Professor Ramarao. He is a chief guest today for the international conference. He has completed 3 PDF in USA and he received many awards, especially Best Research Award by GITM University, and he is highly qualified. In 1998, he cleared a SLED state level eligibility test, and he received a UGC, JRF as well as SRF in 1995 to 97, Junior Research Fellowship by UGC, University Grant Commission, New Delhi, and SRF Senior Research Fellowship, University Grant Commission, 1997 to 2000. And his membership in various academic body, and he review many papers, reviewer in many journals, and member of editorial board like International Journal of Bioassay, International Journal of Chemistry and Life Science, and in the research specialization of Professor Ramarao, medicinal plants, isolation and evolution of anti-cancer, antioxidant compounds, signal transduction, molecular biology, tumor marker, molecular pathway, gene therapy, and hands-on training like molecular biology, cancer biology, protein expression, chromatography, electrophoretic technique, spectroscopic techniques, immunological techniques, animal studies, microbiology, and other techniques. He is well versed in lot of techniques, and he is presented and published paper in a reputed journal, national as well as international, 38 international as well as national journals. He earned the score of impact factor 52 points. It's very high when compared to other researchers, and he participated and presented papers in national as well as international conference, 24 conference, and he invited lectures 6. With these few words, I welcoming sir, Professor Ramarao. Very good morning, sir. We invite Professor Ramarao to present the gathering. Thank you, sir. Thank you, sir. Thank you very kind of. Thank you. Thank you. Professor Ramarao, please stay on the line. Please deliver.
Unmute, Professor. Unmute.
Sir, Professor Ramarao, please unmute and present. Professor Ramarao, please unmute and present, sir. Unmute பண்ணிட்டாங்களா? Unmute பண்ணிட்டாங்களா?
நீங்க வெளில போயிட்டு உள்ள வாங்க.
Yeah, sorry. Yeah, thank you so much for giving me an opportunity. Let me thank your patron RVG and the Punni Madam, and also organizing secretary Muthulingam sir and Malar Selvi for giving me an opportunity to interact with you. Basically my research and postdoctoral research on cancer biology, but they are aptly relevant to this today's seminar or webinar virtual conference on recent innovations of biomaterials in the current scenario, organized by the Department of Zoology and Government Arts College. And today's topic is on biomaterials for cancer diagnosis and drug delivery treatment. You know, the biomaterials, if you look at the history, as like a science, it's very old, more than 50 years old. The biomaterials are the substances that are engineered or natural that interact with many biological systems, especially for the biomedical purpose. These biomaterials either can be used as a therapeutic agents or for the diagnostic purpose. But there is a separate branch for studying these biomaterials. We call it as biomaterial science or biomaterial engineering. These are highly evolved and currently very highly appreciating the branches of these biomaterials. The biomaterials including several metals, ceramics, glasses, and polymers. So currently they are in use for making contact lenses, pacemakers, heart valves, orthopedic devices, and many more. So they may be a natural origin or the synthetic origin. But we know that the biomaterials have a long history. They are in medical applications to support or enhance or replace damaged tissues or biological functions. If you look at the history, first historical use of these biomaterials begins with the ancient Egyptians. They used the animal's tendon or sinew as a sutures. Apart from that, the biomaterials also used for tooth filling. Later on, they are in practice for joint replacement and repair of the hips or cardiovascular repairs. So there are, if you look at the advances, there are advances like any other field, even in the biomaterials also. There are 3 different generations of biomaterials. The first generation biomaterials are synthetic origin. They are biologically inert or nearly inert materials. Originally, they are used to reduce corrosion or to reduce the release of ions. With this objective, they prepare, and also they prepare to minimize immune response when they are implanted into the human body. Because, you know, they are treated as a foreign, mostly our immune system respond and reject during the implantation. So therefore, the first generation materials with the objective to reduce the corrosion and the release of ions to minimize the immune response, they are prepared. There are many examples comes under this first generation biomaterials, like a metals. We know that stainless steel, cobalt, chromium based alloys, titanium or titanium alloys, and ceramics. They're made with alumina, zirconia, and silicon rubber, acrylic resins. They all comes under the first generation biomaterials. Look at the second generation biomaterials. Mostly they are bioactive material derivatives. For example, bioactive glasses, glass ceramics, and calcium phosphates. They are the second generation biomaterials. In mid-80s and 90s, they're very popular and very much used in those times for various biomedical applications, including the polymers, biodegradable polymers. Mostly they are synthetic or natural origin, like a polyglycolide or polylactide. They are extensively used biomaterials for various biomedical applications, even for the nano delivery of the drugs. They are extensively there in use. Coming to the third generation biomaterials, they are prepared with an objective and from the bioactive compounds. With an objective of that bioabsorbable and as a temporary 3-dimensional porous structures, which activates the genes and stimulates the regeneration of living tissues when they are implanted into the human tissue or human body. So therefore, they are not recognized as a foreign because they are easily prepared from the bioactive compounds. And apart from that, the objective in this third generation bioactive materials, they are the combination of bioactivity and biodegradability. You know, they are slowly degradable over the period of time. So therefore, they are not causing any side effects when they are implanted. This is the most important characteristic of this third generation biomaterials. They including alumina, zirconia, and even carbon. The best material, which is mostly used in the third generation bioactive materials, is the carbon. They have a minimal biological response. So therefore, immune system is not responding not inducing any immune response and producing any undesirable effects through the immune system. They're mainly suitable for the orthoprosthetic joints applications to repair the hard tissue in a non-porous form. So these are the third generation, the biomaterials. But like anything, even the biomaterials use also several challenges. So what are the challenges in the 21st century of these biomaterials? One important one is regeneration of the tissue. As told you, when this is implanted, around these implants, the tissue must be regenerated by inducing the expression of the genes around it. This is the most important challenge when you are designing or developing or planning to design or develop a biomaterials. It must be considered that whether this biomaterial is causing regeneration of the tissue around the implants or the devices. That's most important, the challenge. And another one is genetic control of cellular response. It should not be non-toxic. It should not be toxic. It should be non-toxic. And many cellular responses, apart from the immune system, they are induced by these biomaterials. But how to control this cellular response is another important challenge. And we know that we can prepare the hard tissue from these biomaterials, but there are many soft tissues even today we required for biomedical applications. For example, lung or a brain, there are soft tissues they are having. But preparing these soft tissues using biomaterials is another important challenge. And you know that stem cell engineering, they have the self-renewal capacity they have. They can regenerate same kind of the cell and they can differentiate into any kind of the other body cells. But this engineering of the stem cells is another important challenge. Yeah, another important one is when you are implanting these biomaterial devices, or the devices are any items that are prepared from the biomaterials, they may cause some infections. By the infections, because of that hygienity when you are applying these biomaterial devices. How to control such an infection is also another important challenge. And most importantly, the in vitro tests to evaluate the toxicity, to evaluate biocompatibility of the biomaterials. That's very, very important, the parameter, that is important factor that is required, because any biomaterial that is used should be non-toxic to all the bodies. So we should have a methodology or methods or testing methods for the toxicity of the biomaterials before using for making any devices. That's very, very important. And also whether the biomaterials are biocompatible with the body without inducing any immune response. So that is also, there are the important challenge, but we can recall our great scientist, Sir C.V. Raman, with this regard, because based on this principle, there are many methods are today available that called bio-Raman test methods. They're used to evaluate the in vitro toxicity of the biomaterials and also bioavailability of the biomaterials. So if anybody wants, we can refer a journal that Royal Society of Interfaces published in 2010 with this information about the challenges of the biomaterials. That's very, very important. Some of the genes that are upregulated or activated by the biomaterials, especially bioactive glasses. For example, transcription factors and cell cycle regulators. There are many transcription factors, we know that, and also cell cycle regulators like a checkpoint, cycle-independent kinases, and P53 protein, retinoblastoma protein. They're all the cell cycle regulators. They're all upregulated by these biomaterials. Even signal transduction molecules, they're also increased several fold by the many biomaterials. And like the proteins involved in DNA synthesis, the proteins involved in a repair mechanism, the proteins involved in the recombination mechanism is also increased several folds. When growth factors and the cytokines that also increase by 2 to 3 fold by these biomaterials. So, and also some cell surface antigens and receptors, extracellular matrix proteins like a matrix metalloproteinases, MMPs, they're also upregulated. And some of the biomaterials are known to regulate apoptosis or programmed cell death, one of the key cellular mechanism during the treatment, chemotherapy or radiation therapy or immune therapy. So therefore, we must consider when biomaterials are used for the therapeutic purpose, whether they are inducing the upregulation or the activity of the some of the genes which are vital for the biological functions. So look at this. Now coming to the biomaterials for the therapeutic applications or diagnostic applications. Now cancer therapeutics, including the preventive medicine or the diagnosis, different kinds of therapy like radiation therapy, chemotherapy, immune therapy, photodynamic therapy, and even the surgery. But recently, the biomaterials, they're also using for the delivery of the drugs, which is one of the most important requirement for the chemotherapy, because many cancer therapeutics are failing because of the lack of a good delivery system. And also some diagnostic materials, they are delivered successfully to the tumor site by means of the biomaterials. They're also very important in studying the cancer therapy, especially in vitro models. For example, 3D models, we can use the biomaterials to prepare the 3D models. These 3D models mimics the natural conditions or natural tumors in a patient. But what today we are using 2D models, they are not 100% mimicking the original tumors. So therefore, like a 3D tumor spheres, their models are very much important to design and development of the drug delivery agents. They're also required for studying the efficacy of various kinds of therapeutic agents. So therefore, the biomaterials are very important in the development of the chemotherapeutics or immunotherapeutics, etc. So like see here, these are the smart biomaterials for the different applications in the human. So before going to talk about how these biomaterials or the nanomaterials are useful in cancer treatment or diagnosis, I would like to present a few characteristics of the cancer cells. And many of you might be aware of this, but most of the students must know this. A cancer is an abnormal disease with the abnormal cells. They are abnormally dividing without any control. They are destroying the body tissue. The cells which are dividing uncontrollably without any control are destroying the body tissue. They are called cancer cells. Sometimes we may use a different terminology to represent the cancer. One such is a tumor. A tumor is a clump or a mass of a neoplastic cells or cancerous cells. These tumors are two types, the benign tumors and malignant tumors. The benign tumors are not harmful, mostly. They are not metastatic. They localize to the specific area. That's the reason we call them as non-cancerous, because of the thick cutaneous layer over the benign tumors. Unlike the benign tumor, if you look at the malignant tumor, they are highly cancerous. The cancer cells spread to the nearby tissue or spread to the distinct organs in the body. That process we call it as metastasis. It is one of the pathological condition. If the patient is advances, the metastasis happens that affecting the other body parts. But the tumor cells are not new, they are not created, but they are originated from the existing tumor tissue. I mean the tissue of the body or cells of the body. One important phenomenon that is increasing the metastasis and increasing the pathogenicity is the formation of new blood vessels around the tumors. We call this process called neovascularization or angiogenesis, which is a prerequisite, which is essential for a growing tumor, which supplies the oxygen, which supplies the nutrition. So thereby tumor is spread to the nearby tissue or the distinct organs. But the early diagnosis is useful for successful treatment of the cancer therapy. So there are many biomaterials which are currently testing for anti-cancer formulations, like a methods like a co-solvents. There are many advantages, disadvantages with each of these methods: liposomes, polymer mycelium, hydrotropic polymers, lipid emulsions, nanocrystals, and solid dispersion methods. There are the different methods that are used for the preparation of anti-cancer formulations. They have their own advantages and their disadvantages. But one should not underestimate that the biomaterials are a very important role in the making a good formulation with high bioavailability and biodegradability, and especially the tumor targeting the properties. But the materials used in it should be some of the poorly soluble drugs. We know that we have excellent drugs like a paclitaxel, docetaxel, but there are disadvantages with this kind of chemotherapeutics. One is the solubility. So we can improve the solubility by mixing the drugs with the biomaterials, so that we can administer these biomaterials along with the drugs by intravenously. So, and also when these biomaterials mixed with the drugs, they are highly soluble in water. So we can make them by solubilizing by precipitation methods, by precipitating the poorly soluble drugs in the water by applying the biomaterials. But the liposome, the biomaterials which we can use for preparing the liposome formulations, they are widely used for delivering poorly soluble drugs. Similarly, liposomal formulations, already they are in practice, they are in use, especially for delivering doxorubicin. When the doxorubicin is used in the liposomal formulations made from the biomaterials, that reduced the cardiotoxicity, which is one of the major side effects of the doxorubicin. If the doxorubicin is used without a biomaterial, it is highly toxic, and especially it affects the heart. But when the doxorubicin is used in a liposomal formulation made from the biomaterials, that showed the reduced cardiotoxicity. So this is the very important applications. So there are many, the polymer, along with that, the polymer mycelium, they are also widely used for delivery of the poorly soluble drugs. Biomaterials for the making the polymycelium, like polyethylene glycol or polylactic acid, glycolic acid. They're already, as I told you in the beginning, they are some of the biomaterials. They are widely used for drug delivery agents. But what is the advantage of this polymer mycelium made from the polyethylene glycol or polylactic glycolic acid, they are unstable in the blood. So therefore, they are easily disintegrated or disrupted in the bloodstream and releasing the drug. So that's the advantage of using the biomaterials for the making the mycelium for the poorly soluble drug delivery. So apart from that, these nanomaterials, they're made from the biomaterials. They're used in the drug delivery or imaging, and together therapy as well as drug delivery, we call it as theranostic applications. So you may use these biomaterials or nanomaterials for the drug delivery and for the treatment purpose by delivering the different kinds of chemotherapeutic drugs, immune therapeutic drugs, or gene therapy like that. And as I told you, tumor imaging is one of the most important and challenging. If the tumor is detected at the early stage, it can be curable. But if it is detected at a later stage, it is highly impossible for the treatment, for the elimination. So therefore, the biomaterials, we can use it for as a contrasting agents for diagnostic methods like a radiological methods. We can use them as a contrasting agents. Similarly, some biomaterials we can use for both therapy as well as the diagnosis simultaneously. We call it as a theranostic applications of the biomaterials. So have the nanotechnology have a promising applications, especially in the diagnosis and the treatment of a cancer through targeted delivery of their drugs or genes or proteins exclusively to the cancer cells by limiting normal cells or healthy cells. So there are many biomaterials they are used currently for making the liposomes, for making the lipid nanoemulsions, making the lipid carriers and solid lipid nanoparticles, nanostructured lipid carriers, and quantum dots. So look at the liposomes. The liposomes are self-assembling, the spherical, closed colloidal structures. They're made up of lipid bilayer that's surrounding a central aqueous space. Look at here. So this is the advantage of this using liposomes. Here it is in a aqueous media, and inside that, and there is an hydrophobic environment in between this lipid layer. So therefore, we can use the liposomes for the delivery of both hydrophobic drugs. Here, look at here, and hydrophilic drugs. So there's simultaneously, because currently the combination therapy is in practice for treatment of many advanced cancers. So these drugs may be, one may be the water soluble drug, the other one is the water insoluble drug. But both of them are effective and reducing the concentration of the drug required if they are used in the combination. So therefore, we require a drug delivery system that can deliver both hydrophilic drug and hydrophobic drugs simultaneously. So that liposomes have such an applications. They improve the pharmacokinetics. They improve the pharmacodynamics of such a hydrophilic and hydrophobic drugs. Currently, there are many liposomal based formulations there under the use. For example, sheath liposomal doxorubicin, Myoset, they are the trade names. Liposomal donorubicin, that is called donoxomil. There are the different trade names. They are currently there in the practice for the delivery of the drugs. So nanoemulsions, the other kind of the biomaterials are used for making the nanoemulsions. They are the small droplets. They are kinetically stable colloidal systems when compared to the liposomes. So therefore, they have advantages over the liposomes. So because of their inherent property They improve the drug delivery. So they are used for the targeted drug delivery of various types of cancer drugs. And simultaneously, we can deliver anti-cancer drug as well as photosensitizers. So therefore, we can use the nano emulsions in the photodynamic therapy. You know, photodynamic therapy, when the photosensitizer is delivered to the tumor site and exposed to the light, the photosensitizers release the hydroxyl radicals like a superoxide, hydroxyl radicals, singlet oxygen, hydrogen peroxide. They're highly toxic and they damages the tumor tissue. So we can visualize such a tumor even by using photosensitizers. And by using liposomes, nano emulsions, currently the paclitaxel, you know, paclitaxel is one of the first line chemotherapeutic drug, especially for the treatment of the breast cancer and other female cancers like ovarian cancer, endometrial cancer. The paclitaxel is using. But I told you the paclitaxel problem with the paclitaxel is the poor solubility. But if you can use the paclitaxel along with the nano emulsions, it increases the solubility, it increases the bioavailability of the paclitaxel. So it retains in the circulation over long period when it is combined with nano emulsions. So there are other kind of the thing is lipid nanocarriers. They are the nanostructures made with the lipid molecules. They're called lipid carriers. They are produced through controlling of mixing solid lipids with a liquid oil, which leads to the special nanostructures like a matrix. So we can use such a nanocarriers for the delivery of the drugs, as they are new mode of cancer treatment by exclusively delivering the drugs to the tumor site. So therefore, they are the targeted delivery. We can use lipid nanocarriers for the targeted delivery. Another important one is the quantum dots. There is a lot of advancement in the quantum dots. They are highly distinctive size, variable size with the surface characteristics. They have a exclusive properties like a fluorescence because they have a electron gap. Their interaction with the surrounding configuration, they can exhibit the fluorescence. So that we can use the quantum dots to visualize the tumor. Even small, very small tumors can be visualized by quantum dots. So they are acting as a promising platform for biomedical applications. Especially, they can use for the targeted therapy, the targeted imaging, especially for example, sentinel limb mapping. That is one of the most important parameter in the identification of whether the tumor is spread to the other body parts or not. So there also, therefore, quantum dots are using for the sentinel lymph node mapping. So therefore, they can identify even at the early stage of the tumor progression. And even quantum dots can be used as a biomarkers identification, which are very important. The biomarkers, especially non-invasive biomarkers, we can collect the blood and test their blood for some markers. Instead of the tissue biopsy, it is a highly invasive method. So therefore, quantum dots are also using for non-invasive biomarker identification. Coming to the drug delivery, the nanoparticles deliver anti-cancer drug to the tumor by two different mechanisms. So here they exploit either passive targeting method or active targeting mechanism. But the passive target mechanism has the advantage because the tumors have, nanoparticles have an inherited size, very small size, nano size, and also tumor vasculature. The vasculature means the blood vessels around the tumor. Look at here. There are blood vessels which are different from this blood vessel. This is the normal tissue. This is the tumor tissue. One can find the difference. The tumor tissue, the blood vessels in the tumor tissue are highly leaky. They have a small pores. Because of the small size of the nanoparticles, along with the drug, they're escaping from these blood vessels at the tumor site exclusively, but no such a gap or a pore are leaky in the normal tissue. So therefore, the drugs with this kind of the nanoparticles or nanomaterials, they are not escaping at the normal tissue. So therefore, they are not affecting this normal tissue. But because of the leaky of the blood vessels, that inherent property of the any tumor, solid tumors mostly, like a breast cancer, brain cancer, okay, ovarian cancer, they're all these solid tumors, they have a leaky tissue and a vasculature like this. So the passive mechanism exploiting such a leaky tissue to release the drugs exclusively to the tumor site, therefore enhancing the permeability and the retention of the drug, and also the microenvironment, which is made up of the different, other than the tumor cells here, the fibroblasts, immune cells, other also there around the tumor tissue, that we call it as a tumor microenvironment. So this, therefore, because of this kind of the inherent properties, the passive targeting mechanism is highly effective, which enhances the drug bioavailability as well as efficacy. And also, it increasing the long circulating therapeutic nanoparticles, which is possible, and they are passively used in this tumor tissue treatment, and hyperpermeability of the anti-angiogenic drugs that also useful for the treatment of this kind of the drugs. And apart from that, active drug delivery mechanisms, they're exploiting the targeted ligands. Their ligands are linked to the nanoparticles. So therefore, they're exclusively such a antibodies are specific to the cell surface proteins exclusively expressed on the tumor cells. Such a antigens are not expressed by the normal cells. So therefore, this antibody tagged nanoparticles, they are not binding to the normal cells, but because these antigens are exclusively expressed on the cancer cells and antibody tagged nanoparticles bound to the tumor cells or cancer cells, they are internalized like this. So this is the carrier with the ligand loaded with a drug, they're binding to the specific receptor. This receptor drug internalizing by internalization, and finally they're delivered in the cell. So the active drug mechanism has an advantage over the passive drug mechanism. Here, selectively, active drug mechanism selectively deliver the drugs to the only cancer cells, but not the normal cells, because the antigens are exclusively expressed on the cancer cells. So there are coming to the applications of these biomaterials in the cancer cell imaging. I told you, normally the imaging techniques like a CT scan, PET scan, MRI, they are normally used. They are radiological imaging techniques. But the disadvantage with the conventional, these radiological methods is low contrast. So therefore, we cannot differentiate the tumor tissue from the normal tissue. But if you can add contrasting agents along with this radiological method, they can differentiate even small tumors from the normal tissue. Look at here, this is the brain image without a contrasting agent. Entire the tumor is entire the brain part looks like a same. We cannot, even you are unable to see such a tumor, though the tumor is here, but we cannot differentiate the tumor site from the rest of the part. But if the contrasting agents are given intravenously, they distributed over entire the body, even in the brain, because they can cross the blood-brain barrier. You can see that the same MRI with the contrasting agent, we are able to clearly see the tumor. So this is there are different kinds of the contrasting agents, like a T1, T2 contrasting agents, which are used specifically refer to the time that is taken between the magnetic pulses and image that is taken. There are the T1 weighted images and the T2 weighted images. For that purpose, the contrasting agents are normally used. So these are the ultra small iron oxide nanoparticles. They are useful for the contrasting agents for the MRI or PET scan or CT scan. So these are how they are the different size particles. They are helping in imaging the MRI, contrasting agent with the MRI and contrasting agents with the PET scan. We can clearly visualize this. So these are some of the applications. We can also use photodynamic therapy as already mentioned, in which the biomaterials are used. They're causing the toxicity. You know that when the biomaterials as used as a photosensitizing agents, so when this light is exposed, they generate reactive oxygen species that kills the tumor cells. And photothermal therapy, some kind of the biomaterials, they release the heat when they're exposed to near infrared lasers. So when the biomaterials are given and exposed to the tumor site exclusively with the near infrared laser, that generates the heat, which the heat damaging the tumor tissue and causing the necrosis or apoptosis. So in this way, we can also use the photodynamic therapy. And for both therapy and diagnosis, as already mentioned, the quantum dots, they're called as the theranostic applications. They're useful for both treatment, drug delivery, as well as the imaging of the tumor tissue simultaneously. That application, we call it as a theranostic applications. So even biomaterials like a methyl beta cyclodextrin based nano theranostics, they are used for the site specific delivery of the doxorubicin as well as the curcumin. Similarly, another biomaterial, chitosan, the chitosan coated the nanoparticles, they are exclusively delivered the epigallocatechin galates to the liver cancer cells. Similarly, folic acid and glycoric acid conjugated human serum albumin, they effectively deliver the resveratrol to the liver cancer. So therefore, we can use different kinds of the theranostic phytochemical delivery systems from the biomaterials. So from this, I would like to conclude that the biomaterials have several advantages over the traditional treatments of the cancer, like a formulations. They have a potential to carry the drugs exclusively to the cancer cells by using the pathophysiology of the tumor selectively. As I told you, one important one is the tumor vasculature. Similarly, the nanotechnology based drug delivery systems, they explicit the cancer cells, leaving the adjacent normal tissue. That means they target only the cancer cells, but not the normal cells. Even nanosystems exhibit higher the shelf life with a better distribution of the drug throughout the blood. So therefore, the biomaterials used for the drug delivery or the diagnostic purpose and imaging purpose, they're extensively have applications. Thank you. If you have any questions, feel free to ask me, please. So this is about my lab, and these are my mentors, Professor T. Ramana, my PhD mentor, and my postdoc mentor, Chasti Rao. These are the students who completed the PhD or postdoctoral training in my lab. So these are the projects completed or undergoing. These are my collaborators, India and outside the country. Yeah, thank you. If any questions.
Thank you very much, Professor Ramarao, for efficiently, excellently present what is a what is cancer, how it is treated by cancer and diagnosis. I hope all the delegates useful from this lecture and especially the PG students receive the more information regarding cancer treatment and its diagnosis. Thank you very much. On behalf of organizing secretary, we extremely happy and thanks to Professor Ramarao sir. Thank you sir. Thank you very kind of you sir for your
Thank you, sir.
time. Thank you sir. Thank you sir.
Right. So I have a class. I'm leaving, sir. Thank you.
Thank you very kind of you sir. Thank you sir. Okay. Please.
Thank you, sir. Thank you for your excellent talk. All the participants, it's a lunch break for all the participants. We will assemble at 1:30 p.m. sharply. The attendance is very important, so all must attend the program without fail, so that we can give the certificate. So we need your cooperation. We will assemble at 1:30 p.m. in the same Google Meet. Thank you very much. Thank you. Thank you.
Thank you very much. Thank you very much.
நான் இதுக்காக அந்த மெல்லடி
Hello, Mani. சார் சொல்லுங்க. Good afternoon, one and all present here. We start the afternoon section. Today's invited talk is by Dr. K. Krishna Chaitanya. So we request sir to present the invited talk. Welcome, sir. Welcome. Hello. Hmm? Hello.
Professor Krishna.
இருக்கும்னு நினைக்கிறேன் சார் ஃபோன் பண்ணுங்க. Hello, sir. Good afternoon. Hello. Unmute लड़का ने.
Professor Krishna.
Professor Krishna.
Are you audible? Are you audible?
Yeah.
Yes.
Okay, sir. Good afternoon. Shall we start the afternoon session? We kindly invite you to present the presentation, sir. Thank you very much.
Sir, are you getting PPT?
Hello.
Madam, did you get the PPT?
Professor Krishna.
Yes, sir. Yes, sir. Are you receiving?
Okay. Okay, you start the
You start
presentation.
the presentation.
Yes.
Please go ahead.
Sir, it is okay, sir. Yeah, it is okay.
Yes. முடிச்சிங்களா?
Sir, it is okay or not?
கனெக்ட் பண்ணுங்க காலையில உள்ள.
Yes. Very good afternoon.
Okay, proceed, sir.
Yes, sir. Yeah, a very good afternoon to everyone. Okay, thank you for your invitation. Now I'm talking about evolution of anti-inflammatory and cytotoxic activities of Trimenia brownie silver as well as zinc knot particles synthesized by the medicinal plant. You see, first of all, we are going to inflammation. Already morning we discussed about the inflammation. What is inflammation? We are having the two different type of inflammations are there. One is acute inflammation, another one is chronic inflammation. In chronic inflammation, inflammatory mediators are upregulated. These upregulated inflammatory mediators are responsible for different inflammatory related diseases and cancer. Okay. Yes, we are currently targeting chronic inflammation by targeting different inflammatory mediators like 5-LOX, COX, and induced nitric oxide, and some inflammatory mediators like pro-inflammatory mediators like interleukin 1, interleukin 6, nitric oxide, and anti-inflammatory like interleukin 10 and interleukin TGF beta. Okay. Just for controlling chronic inflammation, we are using both steroidal anti-inflammatory as well as non-steroidal anti-inflammatory drugs. Okay. But you see, these anti-inflammatory drugs having some side effect. That's why we are focusing developing of the novel drugs by using green synthesis nanoparticle. You see, both steroidal and non-steroidal anti-inflammatory drugs are bulky in nature.
Sir, excuse me.
Yes, sir.
Kindly show your screen. Screen is not presenting.
Not presenting?
Yeah.
Okay.
PPT, PowerPoint not visible.
Yeah.
Please show your PowerPoint also.
Sir, it is share is coming, sir. Share.
Krishna.
Yes, sir. Sir,
Please
share.
show your PowerPoint.
No, sir.
Then only the audience will can able to find out your voice not at all clear.
Yes, sir.
Please try to present with PowerPoint.
Okay.
Thank you.
Yeah. Sir, now we are getting share. Are you getting now? Hello, sir. Are you getting?
Sir,
Are you getting?
sir, the
Are you getting?
PowerPoint presentation, please share your slide.
Where, sir? I am sharing, but it is not coming. Where, sir? Where I have to share?
Krishna, please share your slide.
Yes, sir. Yes, sir. Yes, sir. Yeah. No, sir. Where is the option for sharing? It is not showing any option. Sir, it is not showing any option, sir.
அவர் காமிச்சிருக்காரா ஆப்ஷன் இல்லை அப்படிங்கிற மார்னிங் யூ ஆர் பிரசன்ட் வித் ஸ்லைட்ஸ் நோ
Yeah, sir. Yes, sir.
லைக் தட் டூ சார்
Okay. 1 minute, sir. 1 minute. Now, sir. Now, what about now?
because you are presenting far away, sometime your voice may be break.
Okay.
So with the help of PowerPoint only
Yes, sir. Yes, sir. Yes, sir.
the
Yes, sir.
audience can able to understand what you said.
Okay.
Please try to place your PowerPoint.
Okay, sir. Wait, sir. Wait.
Okay, continue.
Yes, sir. Sir,
Okay,
where,
sir, continue, sir.
Sir, any options are there, sir? Option for sharing?
Here you only play, sir.
Yeah. I'm keeping, sir.
Option இல்லை அப்படிங்கிற மாதிரி
இல்லை அவங்க ஓபன் பண்ணு.
Okay.
Krishna, you only present the table. Please show your slide.
Okay. Sir, what about now?
Krishna, not visible your PowerPoint.
Yeah, wait, sir. Wait 1 minute, sir.
International conference.
Yeah, wait, man. Wait, yes, sir. Wait. On, madam. ఎక్కడ మేడం Sir, share is not coming, sir. Sir, wait 5 minutes, sir. Wait. Share is not working.
Excuse me, madam. S.A. Krishna Kumari. Please mute your audio.
Yes, ma'am.
Krishna Kumari. Okay, thank you.
Sir, share. Are you getting now?
Krishna, please do one thing. Krishna?
Yes, sir.
Please do one thing. If possible, please kindly send mail your PPT to our mail ID.
Okay.
Research dgggac@gmail.com.
Okay, sir. Okay.
Krishna?
Yes, sir. I'm sharing, sir. I'm sharing.
Kindly
to
send
Yeah.
your
Mail.
PPT.
Yes. రాట్లే షేర్ అవ్వట్లేదు నాన్న.
Krishna?
Yes, sir. I'm sending.
Kindly send your PPT
Yeah.
to researchdggac@gmail.com. Already you know the mail
Okay,
ID.
yes, sir. Yes.
Okay, thank you. Okay.
Sir, check, sir. I send it, sir.
Krishna?
Sir, I sent, sir. Check, sir. 1 second, sir.
Okay. Kindly send it.
Yeah. Sir, got it, sir? Yes, sir. I sent it, sir. Sir, I send it, sir. Check, sir. 1 second.
Krishna?
Yes, sir. Send it, sir.
Thank you. Received.
Yeah.
Received your PPT.
Yes,
Okay,
sir.
thank you.
Yes, sir.
Krishna?
Yes, sir.
Is it okay, sir?
Yeah.
Okay, if you want to move the slide, please tell me we move. Okay?
Okay, sir. Are you getting?
Okay, thank you.
Okay, sir. Yeah. Sir, are you shared? You are shared, sir? Okay. Yeah, first one. You see the title is Evaluation of In vitro Anti-Inflammatory and Cytotoxicity Activity of Green Synthesized Nanoparticle from the Medicinal Plant. First of all, before we have to go for inflammation, we are having two different types of inflammations are there. One is acute inflammation and another one is chronic inflammation. During chronic inflammation, we are having some inducible enzymes are there, and the LOX, COX as well as activated. These activated inflammatory enzymes are responsible for progression of inflammatory related diseases as well as cancer. Currently, chronic inflammation is chronic inflammation is controlled by both steroidal as well as the non-steroidal drugs. These steroidal as well as the non-steroidal drugs having some side effect. That's why we are focusing development of the novel drugs for treatment of inflammatory related diseases as well as cancers. You see, currently the scientists, they're focusing on novel drugs, these novel drugs which are having more advantages because both non-steroidal as well as the steroidal drugs having bulky nature. That's why we are unable to get a good result from by using inflammatory enzymes. That's why currently the scientists focusing on the nanomedicine. In the nanomedicine, the scientists, they prepared different nanoparticles by using physical methods, chemical methods, as well as the biological methods. I am selecting your biological methods to improve the drug accessibility to tissues. You see, nanoparticles are this very small molecule. Okay, they are responsible for decreasing inflammation by reaching the target. You see, currently nanoparticles produce different sizes of nanoparticle, different sizes and morphology of nanoparticle with different physicochemical electrical properties. These nanoparticles having the multiple applications in the field of medicinal sciences like diagnosis and scanning as well as the treatment of diseases. Because here important thing is we are preparing nanoparticle to control the side effects of bulk drugs like both steroidal as well as non-steroidal drugs. In present study, we are synthesizing different nanoparticle by using medicinal plants. Nanoparticles are prepared by different metals. We are having the metal-based nanoparticles or metal oxide nanoparticles. These nanoparticles are having important in diagnostic, therapeutic as well as the drug delivery system. See, we are having two different types of nanoparticles are there. One is metal nanoparticles, another one is called metal ion nanoparticles. These nanoparticles having a diversified biological property, they're responsible for a drug delivery, imaging of the tumor and inflammatory regions of the tissue damaged area. You see, you know very well, nanotechnology is the science. Nanotechnology is nothing but a synthesis of the molecule at nano level. Nanoparticles are, we are having the three different type of nanoparticles are there, biological nanoparticle as well as the chemical nanoparticles are there. These nanoparticles, minimum it contain the 1 to 100 nanometers in size, which are having different physical, chemical, mechanical as well as biological property. When going to synthesis of the nanoparticle, we are having the three methods of synthesis of the nanoparticles are there. One is physical method, chemical method, another one is called biological method. In chemical method, we are using laser beams, radiation as well as electrodeposition, because these physical methods are majorly used in engineering technology for production of different nanoparticles used in different diagnostic tools. As well as in chemistry people, there are also used the three different methods. Among the so many methods, three methods are very important: Sol-Gel method, redox process method, and hydrothermal method. But as a biologist, we are having some disadvantages in physical as well as the chemical method. That's why we are focusing on a synthesis of the nanoparticle by using the biological material, because biological material means we are using different by using animal products. Also we are preparing nanoparticle. By using plant products also we are getting nanoparticle. For microbes also we are getting nanoparticle, because here majorly we are green synthesis means synthesis of nanoparticle by using the plants and by using the blue-green algae is called green synthesis. Because in biological synthesis we are having the different biological products are responsible for the synthesis of the nanoparticle, but in green synthesis we are using plants as well as the microorganism. You see, that's why we are having how many methods are there: physical method, chemical method, as well as the green method, green synthesis. In green synthesis, by using the plant extract, we are synthesizing nanoparticle, because the plants which containing medicinal plant containing the secondary metabolite, here very important thing, these secondary metabolites are responsible for synthesis of nanoparticle, and these plant secondary metabolites also act as medicinal value. Because you see, these phytochemicals which are act as reducing agent and capping agent and stabilizing agent for synthesis of the nanoparticle. Because you see, we are having the three different methods of nanoparticles are there: physical, chemical, as well as the green synthesis. See, green synthesis is having more advantages are biocompatible in nature, highly stable nanoparticle, non-toxic in nature, and broad therapeutic index, as well as biodegradable. These are the major advantages of green synthesis nanoparticle. Here you see, very important thing is Why we are going green synthesis? Before going to the further process, first we have to discuss about nanoparticle. What is the meaning of the nanoparticle? Nanoparticles are nothing but having one dimension with a diameter of 1 to 100 nanometers. Because you see, these 1 to 100 nanometer diameters are there. For synthesis of these small size nanoparticle, okay, we need some reducing agent. We are getting reducing agent from the chemicals by using the chemical method. But here our during green synthesis, these secondary metabolites having major two advantages. One is these secondary metabolites act as a reducing agent, which decreases the size of the nanoparticle, size of the material into nanoparticle. We are having the two different type of synthesis are there. One is called bottom up as well as the bottom down. Here we are synthesizing nanoparticle by using the bottom up method. In a bottom up method, we are taking the plant extract. These plant extract containing the different secondary metabolite. These secondary metabolites act as reducing agent as well as the stabilizing agent. These stabilizing and reducing agents responsible for synthesis of the nanoparticle, which are having the biocompatible in nature, highly stable in nature. You see, already we discussed both physical and chemical methods having some disadvantages. To overcome these disadvantages, scientists they focus on synthesis of nanoparticle by using the medicinal plants. It's called green chemistry. You see, here we are having the three methods of synthesis are there. One is physical method, chemical method, biological method, and after the synthesis of nanoparticle, okay, nanoparticle by the bottom up method. What is the meaning of bottom up method? Means formation of the nanoparticle from the minute molecules. It is called what? Bottom up method. After synthesis of the nanoparticle, those nanoparticles are the characterized by the different technique. These characterization technique are responsible for to confirm the formation of the nanoparticle at initial level to at a multiple level. This is first of all important thing. We are having the different characterization techniques are there. The simple characterization technique is color changing. When during formation of nanoparticle, colors are different colors are formed. Okay, because while synthesis of the nanoparticle, we are taking different metals as well as the metal oxide mixing with different plant extract. After mixing the plant extract with the different metals and metal oxide, we are getting different colors. The based upon their colors, we are confirming the nanoparticles are formed by the process of phytoreduction. After the color, based upon different nanoparticle exhibit different type of the color. Based upon the color also we have to confirm, but that the preliminary, and after the formation of these nanoparticles, we are going for the different technique like UV spectroscopy, FTIR, XRD, DLS, and SEM, as well as the EDX and TEM. Okay, these are the techniques which are responsible for to confirm the nanoparticle based upon the properties of the nanoparticle. I already informed you nanoparticles having different physical, chemical, as well as the electromagnetic properties. All these properties are characterized by using all these technique. Okay, different nanoparticles exhibit different UV, and different nanoparticles also containing different functional group. And XRD responsible for to explain about the crystal structure of nanoparticle, and the DLS. You see, we are a very important thing is UV visible spectroscopy, which confirm the type of nanoparticle, either silver nanoparticle, zinc nanoparticle, or gold nanoparticle. And what is important? The FTIR. FTIR, you know very well, which is useful for identification of the functional group. You see, while preparation of the nanoparticle, we are using plant extract. We are using the plant extract. These plant extracts are mixed with the metals as well as the metal oxide. You see, these plant extract contain the different secondary metabolite. These secondary metabolite contain the functional group. These functional groups are responsible for the reduction of metals and metal oxides into metal oxide nanoparticle. We are confirming the different functional group by using the FTIR. Another important thing, after the formation of the nanoparticle, important thing is the characterization of the crystal nature of the nanoparticle will done by the XRD based upon their Bragg's equation. You see, while you are preparing nanoparticle, we are getting different sizes of the nanoparticle. The sizes ranges from the 10 nanometer to 100 nanometer. That's why while preparing the nanoparticle, we are getting different sizes. Those sizes are analyzed by the DLS, dispersive light spectrum, as well as surface charge also identified. We are having, while preparation of nanoparticle, we are getting two different charges are there, positive charge as well as the negative charge. These charges of nanoparticles are also analyzed by the DLS. And the SEM which is useful for here, very important thing is morphology of nanoparticles are very important to take our drug and to target into the targeted area like cancer as well as inflammatory tissue. That's why the morphology of the nanoparticles are analyzed by SEM. And you see charge of we are preparing nanoparticle by using the metals. These metal composition will be determined by the EDX. Then internal structure of nanoparticle, like internal structure of nanoparticles are determined by the TEM. That's why all these techniques are responsible for to confirm the formation of nanoparticle, to characterize the nanoparticle morphology, size, as well as distribution. You see, already we know we are discussing about metal and metal oxide nanoparticle because in my proposal research work I prepared the both metal as well as metal oxide nanoparticle. Like I prepared the silver as well as silver nanoparticle, copper nanoparticle, as well as zinc oxide nanoparticle. That's why in my present titanium dioxide nanoparticle also. That's why because my students, they are working on different nanoparticle like silver, zinc, gold nanoparticle and titanium dioxide nanoparticle. Here I am explaining about only silver nanoparticle as well as zinc oxide nanoparticle. Silver is metal and zinc oxide is metal oxide nanoparticle. Because why I selected two different, one is metal, one is metal oxide, because based upon their property. Because you see, these metal and metal oxide, it is having different property, because you know very well nanoparticles having very small, because when it is small, it is having large surface area and volume ratio, and it is reduced morphology and large surface energy. Okay, while we are preparing nanoparticle, we are getting all characteristics. You see, here very important thing is currently we are having different inflammatory drugs as well as the cancer drugs are there. But those drugs which are having the bulk in nature, when it is bulk in nature, it is unable to penetrate to tissue and it unable to enter into the target region and it unable to cure the disease. That's why our major intention is we are preparing nanoparticle. We are preparing nanoparticle because bulk means we are having the disadvantage. That's why these bulks are break down into small nanoparticles. Okay, that is also called that is called another breakdown and bottom down effect. Bottom up as well as the bottom down. You see, that's why our metal and metal oxide nanoparticle having large surface area, reduced morphology, large surface energy, and these properties we are getting while preparation of nanoparticle. These characteristics are analyzed by using the different technique. Because these metal and metal oxide nanoparticle used in the biomedical field for act as a drug carriers and treatment of the diseases and the monitoring of the cancers as well as monitoring of cancers while the scanning and prevention of both cancer as well as inflammatory related disease. Not only cancer, we are targeting different diseases. And you see, our main statement of the problem, we are important thing is we are preparing nanoparticle by using the physical as well as the chemical as well as the biological method. In physical chemical method, we are having disadvantages. But among the already we discussed about the disadvantages in previous slide. But here very important disadvantage is size. And during physical and chemical method, we are not getting our accurate size. Maybe we are getting macromolecule. We are unable to get the micromolecule. That's why our major intention is we are getting a unique size of nanoparticle with unique morphology and size range. You see, that's why physical and chemical method they produce the nanoparticle because those sizes or shapes are undesirable in biomedical sciences. That's why we are preparing nanoparticle by using the green synthesis because here so the size, the size and shape and distribution of the nanoparticle depend upon their concentrations. Here we are using a different we are go for the standardization. While the standardization, we are taking different concentrations of plant extract mixed with different molarities of silver as well as the zinc oxide nanoparticle. The based upon the standardization, we are getting 0.1 molar zinc oxide zinc estate useful for synthesis of nanoparticle. That's why you see, while you are using physical and chemical approaches, we are not getting our desirable size of nanoparticle. That's why we are focusing on the green synthesis of the nanoparticle. Another important thing is these nanoparticles also, when reach a target area, also it is interfering with the different biochemical reactions of a metabolic reaction. Those reactions also causing disadvantages. Another important statement of the problem, we are having the non-steroidal anti-inflammatory drugs. These are causing some toxicity. It is causing toxicity to different organ, and that's why by using the green synthesis, we are we are overcoming the toxicity. We are overcoming the toxicity of both physical as well as the chemical method. That's why we are synthesizing we are synthesizing green synthesizing nanoparticle by using the silver as well as the zinc oxide nanoparticle by using the medicinal plant for treatment of inflammatory related diseases. These green synthesizing nanoparticle having biocompatible nature, non-toxic in nature, used as a drug carrier in the field of biomedical sciences. And you see, an important thing is significance of the study. You see, we are preparing nanoparticle by using the medicinal plant. Okay, after while preparation of first we have to select the medicinal plant based upon the tradition important. After the selection of the medicinal plant, we can go for the phytochemical analysis, preliminary phytochemical analysis, qualitative, quantitative, as well as GCMS. While these phytochemical analysis, it gives some information about bioactive compounds are information about bioactive compound. That's why while we're preparing nanoparticle, first we have to analyze the our plant extract for identification of different bioactive compound responsible for the medicinal property. That's why significance medicinal chemistry determines. And you see, and it is also useful for the plant extract which contain different bioactive compound. These bioactive compounds also responsible for to curing the cancer, to curing the inflammation. That's why we are isolation and identification of the bioactive compound also our important significance of the study. And you see, after isolation of the compound and after preparation of the extract, these compounds as well as the extracts are responsible for synthesis of the different nanoparticle. Like here, our major intention is we are preparing nanoparticle by using Trimina brownie, traditionally useful for the treatment of inflammation. That's why we are selecting Trimina brownie. Here we are doing two important. We are measuring two important parameters. One is inflammation, another one is cancer. You see, for inflammation we are having the raw cells or the model cells. For the breast cancer, we are having MDA, MDB cells are the model cells to check the cytotoxic effect of breast cancer cells. That's why by using plant extract, we are preparing the silver as well as the zinc oxide nanoparticle, which are having more biocompatible and eco-friendly and high therapeutic value when compared to the normal chemical drugs. Okay, that's why these are the innovative technique for the researcher who are interested to do synthesis of the nanoparticle. This is the model research work for the people who want to work on green synthesis of the nanoparticle. Then object of the work. Here our important thing, we are preparing two different nanoparticles by using the Trimina brownie, one is silver as well as the zinc. And after the preparation of these nanoparticles, like TB means Trimina bromine, silver nanoparticle, and zinc oxide nanoparticle by using the plant extract. And these nanoparticles are checked for, evaluated for anti-inflammation as well as the anti-cancer activity by using the two different cell line. One is called raw cell line, another one is called MDB cell line. Our objective is to evaluation of anti-inflammatory as well as the cytotoxic effect of green synthesis nanoparticles. It is a general objective. Then we are coming for specific object. When we are going for the specific object, in a specific object, first we have to prepare the aqueous extract of selected medicinal plant. Then go for the phytochemical qualitative and quantitative phytochemical analysis as well as the GCMS as well as the FTIR. And these preliminary and advanced phytochemical analysis responsible for give information about what type of the phytochemicals are present in extract and what phytochemicals are present in extract and what are the possible compounds are responsible for to mediate biological functions like anti-inflammatory as well as anti-cancer. And the first objective is we have to prepare the plant extract and go for phytochemical analysis. And second objective is and the preparation of silver as well as the zinc oxide nanoparticle by using precursors of silver nitrate and zinc nitrate precursor. Here you see we are preparing two nanoparticle. One is silver nanoparticle and zinc oxide nanoparticle by using bulk material. Here bulk materials are silver nitrate is a bulk, zinc nitrate is a bulk precursor. By using these bulk precursors, we are preparing a nanoparticle by using the plant extract. And after the preparation, we are go for the characterization of nanoparticle by using a simple to advanced technique. And first we can go for the phytochemical analysis, then the synthesis of the nanoparticle, then go for characterization. Then we have to check the cytotoxic effect. Check the cytotoxic effect of nanoparticle because you see, for a development of anti-inflammatory compound, our compound should not showing any cytotoxic effect. That's why we synthesize the nanoparticle, and we have to check first of all cytotoxicity. That's one, to evaluate the cytotoxicity. And after the checking the cytotoxicity and based upon the cytotoxic effect, we have selected the nanoparticle to check anti-inflammatory activity in raw cell. And after the checking the raw cell, here a very important linkage. You see the molecule which does not have any cytotoxicity, with showing anti-inflammatory activity. It is very important criteria for to develop anti-cancer drugs. Here, my important thing is important point is, you see, inflammation is protective, but when acute inflammation became chronic inflammation, this chronic inflammation became cancer. Okay, these cancers are responsible for malignant as well, benign tumors as well as the malignant tumor. That's why here we are targeting inflammation, chronic inflammation. When we are controlling chronic inflammation, automatically we are also controlling cancer. That's why my main important goal is we have to develop dual molecule, because our nanoparticle, it acts as both anti-inflammatory agent as well as anti-cancer agent. That's why first I have to check the cell viability, and it should not showing any cytotoxicity against normal cell line. Then I have to go for evaluation against anti-inflammatory. Then I can go for cytotoxic activity. You see, this is my background of the work. In the background of the work, you know very well. You see, first important thing is here I am using the medicinal plants. Here important thing I already discussed about you. My important thing is synthesis of the nanoparticle by using the green synthesis. For the green synthesis, we need plant extract. First, we have to select the plant. Here I am selecting Trimenia brownie. This plant extract is once again authenticated by the botanist and further authenticated by the Central University to confirm the medicinal plant. You see, this medicinal plant here, important thing is this medicinal plant traditionally used for the treatment of the inflammation as well as the cancer. That's why, based upon the traditional information, I collected this medicinal plant from Oxum. And you see, after the selection of the medicinal plant, this medicinal plant is treated with demineralized water, and after that I make into that powder, and after the making into the powder, I make extraction with aqueous extraction. Aqueous means, you see, while preparation of nanoparticle, here aqueous extraction is very important for the preparation of nanoparticle, because you see we are having the 4 different organic extracts also there, and aqueous extracts also there. And here while preparation of green synthesis nanoparticle, better to use ethanol as well as aqueous, because you see for preparation of nanoparticle we are using the bulk salts. These bulk salts are only dissociated in aqueous solutions only. It does not dissociated in organic solvent. That's why here we are using two different metal precursors, silver nitrate as well as the zinc state. These silver nitrate and zinc state easily dissociated in water when compared to the other organic solvent. You see, that's why first we selected the aqueous extract, we selected the medicinal plant, and we make into the powder, and after the make into the powder, and we have to make aqueous extract by using the oxalate extraction. And after oxalate extraction, we can go for the qualitative analysis and the quantitative phytochemical analysis and standard method. And after qualitative and quantitative analysis, we are getting some information what type of the secondary metabolites are present in plant extract, and what type of secondary metabolites are present in plant extract. Because when we are knowing these secondary metabolite, then only we have to confirm that these secondary metabolites are responsible for formation of nanoparticle as well as showing anti-inflammatory as well as anti-cancer activity. And after synthesis of the aqueous extract, we can go for synthesis of silver nanoparticle, zinc nanoparticle by the standard procedure. And after the synthesis of the nanoparticle, we should confirm the nanoparticle by using the color change, by using a visible absorption and optical as well as the morphological as well as the structure as well as the morphological changes as well as chemical composition by using the different advanced technique already I discussed before. And after the synthesis of the nanoparticle, the initial step is here. First, our nanoparticle, it should not show any cytotoxicity. That's why we are preparing the 2 nanoparticle like a silver nanoparticle and zinc oxide nanoparticle. These nanoparticle, it does not show any cytotoxicity. If it's showing cytotoxicity, it does not use it for anti-inflammatory drug. That's why first we have to check the in vitro cytotoxicity by using the 2 nanoparticle. Here control is active aqueous extract. That's why we have to take the 3 sample. One is TBAQ, aqueous extract of terminobroni, the silver nanoparticle, and zinc oxide nanoparticle. These 3 sample, 2 samples with aqueous extract treated with raw cells to check the cell viability. And based upon the cell viability assay, we have furtherly carried out anti-inflammatory activity by taking 2 reference molecule nitric oxide as well as the PGE2. After conducting the anti-inflammatory activity, we should take we have to take the breast cancer cell line. These breast cancer cell line treated with different concentrations of the selected nanoparticle. First of all, that's why first we have to go for extraction, and after extraction we have to prepare the nanoparticle. First we can go for the extraction, aqueous extraction, and after that, according to the protocol, first selection of the medicinal plant is important. After the selection, we can go for the dry, and after the dry, and we can go for the powder, and this powder is make into the aqueous extraction. This aqueous extract is used as reducing as well as the capping agent for the synthesis of both silver nanoparticle as well as the zinc oxide nanoparticle. That's why this aqueous extract act as reducing as well as the capping agent for the synthesis of silver as well as the zinc oxide nanoparticle. And after the synthesis of these silver as well as the zinc oxide nanoparticle, first we have to check with confirm the formation of the nanoparticle by using the different physical as well as the chemical method. Then we can go for the cytotoxicity, preliminary cytotoxicity, and if it is showing good result with cytotoxicity, but our nanoparticle should not show any cytotoxicity against raw cells. If any nanoparticle exhibiting cytotoxicity, we should not consider those nanoparticles. That's why we have to select the nanoparticle based upon the cytotoxicity, and after that we have to consider, we have to check the inhibitory activity against nitric oxide as well as the PGE2, and after the checking of these mediators, then we can go for the cytotoxic effect. These are the methodologies. I done all preparations based upon the standard procedures. You see here important thing is, important here, synthesis of the nanoparticle. While the synthesis of the nanoparticle, first we have to take the this is the A which show the silver nanoparticle, as well as the B zinc oxide nanoparticle, and the C it is called a solution. Okay, that's why color changes. You see, for example, already discussed with you. You see, color visible observation is very important criteria to confirm the formation of the nanoparticle. Color changes from the color to colorless to reddish brown, and green to dark brown confirm the formation of silver nanoparticle as well as zinc oxide nanoparticle. You see, the first one is aqueous. A is aqueous extract, and the B is silver nitrate as well as the C is zinc oxide nanoparticle. You see, when you are seeing the color, of this plant extract, it is a pale color. But once here we confirm the formation of silver nanoparticle. Once color changed from a colorless to reddish brown color, it confirms the silver, and as well as green to dark brown means because you see reddish brown, it indicates the silver nanoparticle, and the dark brown indicates the silver zinc oxide nanoparticle. That's why the change in the color of the plant extract after addition of silver nitrate as well as the zinc estate, it confirms the formation of the nanoparticle. Within the 30 minutes, we are getting this color change due to the process of bio-reduction. That's why it is called which synthesis? Green synthesis. Here, we take the plant extract. To the plant extract, we are adding silver nitrate as well as the zinc estate, and after adding 30 minutes, while continuous stirring, we are getting 2 different type of the nanoparticles, silver nanoparticle, zinc oxide nanoparticle, and these nanoparticle confirmation will be done by the change in the color. One is reddish brown color for the silver nitrate and dark brown for zinc oxide nitrate. And you see, after the preliminary confirmation, we can go for the usual spectroscopy, and usual spectroscopy because different scientists, they reported that the silver nanoparticles also confirmed by the usual spectroscopy, based upon the standard procedures. Okay, we are synthesized the 2 nanoparticle. The 1 nanoparticle, it is showing the 428 nanometer. Another nanoparticle, it is showing the 294 nanometer. You see, the based upon their standards, based upon their different review of literatures, and based upon their standard values, 428 nanometer. Because you see, already I discussed it to you. Because you see, red brownish color, it shows absorption at 428 nanometer. That's why it is confirmed that formation of silver nanoparticle. In the same manner, you see formation of the dark brown. Also confirm that formation of zinc oxide nanoparticle. That's why these two nanoparticles are having the different color intensities. These two different color intensities is read at two different optical activities. One is 428 as well as 294 nanometer. And you see, here important thing is here you see here while green synthesis of the nanoparticle, the secondary metabolites which are present in the plant textiles are responsible for to reducing the size of the bulk molecule like zinc acetate as well as the silver nitrate. That's why once again we should confirm the presence of the secondary metabolites are responsible for act as reducing agent as well as the capping agent done by FTAR. This FTAR, it gives information about the possible numbers of the functional groups in aqueous extract. You see, here important thing is, you see, the termina broni aqueous extract, it contains the different functional group. After the preparation of, you see, in the second column, silver nitrate, and the third column is the zinc oxide nanoparticle. While you are seeing the differentiation between the aqueous extract and the silver nanoparticle and zinc oxide nanoparticle, some variations are there. Here, during synthesis of the nanoparticle, the aqueous extract loses some functional group. These functional groups are act as reducing agent, as well as the capping agent, and these functional groups are involved in the formation of silver nanoparticle as well as zinc oxide nanoparticle. That's why FTAR confirmed the FTAR report confirmed the involvement of the different functional groups for the synthesis of the silver nanoparticle as well as zinc oxide nanoparticle. You see, this is the peak of termina broglie aqueous extract FTAR. You see, in the FTAR, it contains the different functional group. You see, the different functional group, we are containing the huge amount of the functional group. But you see, when you are seeing silver nitrate, those functional groups are shifted. Those functional groups are shifted to different nanometers. Because what is the meaning? Yes, what it denotes means, what it indicates means, okay, the functional groups which present in the aqueous extract are responsible for the synthesis of silver nanoparticle. That's why some functional groups are missing in aqueous extract. Because missing means those functional groups are involved in synthesis of the zinc oxide nanoparticle. That's why when we are seeing silver nanoparticle, we are missing some peaks. These peaks are nothing but involved in synthesis of the silver nanoparticle. But you see here, the plant extract acts as a reducing agent as well as the capping agent. In the capping agent means the cap, it contains different functional groups. Okay, these functional groups are act as anti-inflammatory as well as anti-cancer agent. You see, in the same manner, zinc oxide nanoparticles also loses some functional groups in the aqueous extract, because it is also containing some silver some of the functional groups are shifted. Some of the functional groups which present in the aqueous extract are shifted towards other functional groups due to the presence of zinc oxide nanoparticle. Because you see, while you are seeing these three FTAR diagram, you see aqueous extract containing the more functional group when compared to the silver nanoparticle as well as zinc oxide nanoparticle. Because due to what it indicates means the functional groups which present in the aqueous extract act as reducing agent and capping agent for the synthesis of the silver nanoparticle as well as the zinc oxide nanoparticle. Then you see, after the first we confirm the nanoparticle based upon their color. After the color, we can go for the usual spectroscopy, and after the usual spectroscopy, we are going for FTAR. This FTAR, it gives what type of the functional groups and involved in the synthesis of the nanoparticle. And after that, we can go for a XRD analysis. This XRD analysis, it gives crystal structure of a nanoparticle. Because you see, our nanoparticle, one of the characteristics is it should contain the crystals. But that's why the crystal nature of nanoparticles, like a silver nanoparticle as well as the zinc oxide nanoparticles, are confirmed by XRD. And after the confirmation of the crystal nature of the nanoparticle, okay, we have to see the distribution. You see, already I discussed with you, while we are preparing nanoparticle, we can't control the size of the nanoparticle. We are getting different sizes of the nanoparticle, which are ranges from 110 to 100 nanometer. That's why while we are preparing the nanoparticle, we are getting different sizes of the nanoparticle. These sizes of the nanoparticles are confirmed by DLS, dispersive light spectroscopy. Okay, while dispersive light spectroscopy, you see very important thing is silver nanoparticle dispersion is ranging from 10 nanometer to 162 nanometer. While seeing zinc oxide nanoparticle, it ranges from 10 to 80 nanometers in the size. That's why while you are seeing, while synthesis of the nanoparticle, we are getting different sizes of the nanoparticle. For the silver, we are getting 10 to 162 nanometers in the size. For the zinc, we are getting 10 to 80 meters in the size. That's why this DLS gives the probability distribution of the nanoparticles in solvents. And after the DLS, we can go for the energy. You see, every nanoparticle it should carry the energy. This energy will be determined by the zeta potential energy. You see, if it is having energy, it is having stability. The stability of the nanoparticles also depend upon their charge. If it is more stable, and it containing the more charge. That's why our synthesized nanoparticle, both silver nanoparticle as well as zinc oxide nanoparticle exhibit different charges. Silver nanoparticle exhibiting -18 and zinc oxide nanoparticle exhibiting -21. It indicates that you see zinc oxide nanoparticles are more stable than silver nanoparticle. Then after the computation of zeta potential analysis, you can go for the SEM analysis. You see SEM scanning. It is scanning. We are having microscopy. Generally, microscopes are useful for identification of the morphology as well as the internal structure. Here we are having SEM analysis. SEM analysis means identification of morphology, the confirmation of morphology of the green synthesized nanoparticle. The first A is the morphology of silver nanoparticle, and B is morphology of zinc oxide nanoparticle. You see, silver nanoparticle shows the polydispersive. Polydispersive means different sizes of nanoparticles are dispersed in the spherical nature. Okay, spherical. But you see, both are polydispersed, because you see, already I discussed in previous slide. I mentioned you, because both are giving different sizes of the nanoparticle. That's why both are polydispersive. But here important thing is silver nanoparticles are spherical in shape, but zinc oxide nanoparticles are pseudospherical in shape. That's why this SEM analysis gives information about shape or morphology of nanoparticle. Okay, from our result, silver is spherical in shape, and zinc is pseudospherical in shape. And after that, we can confirm what type of the metals are useful for the synthesis of the nanoparticle. Here we are having, but we are using two different metals. We are using for synthesis of the nanoparticle, green synthesis nanoparticle. We are using two different metals. One is the silver as well as the zinc. Here EDX it confirms the presence of silver as well as the zinc oxide in green synthesized nanoparticle. You see, the silver exhibit at 3 kilovoltage, and the zinc exhibit at 1 kilovoltage. Here, important thing, EDX analysis means this EDX which is responsible for to gives information about our nanoparticles are made up of which type of the metals, either zinc, either silver, either gold. That's why this is very important step to confirm what type of the metals which present in nanoparticle. You see, after the SEM analysis and the EDX analysis, we should go for internal structure. Here, internal structure will be analyzed by using the TEM analysis. While the TEM analysis, we are getting spherical shape. Already I explained to you. You see, the TEM analysis. You see, we are having the two different shapes are there. You see, silver having the spherical shape, as well as the zinc having the pseudo spherical shape. You see, while you are seeing the TEM structures, in the STEM structure, we are getting a round spherical shaped nanoparticle. Okay. In the same time, we are also getting different sizes of the nanoparticle. You see, we succeed in Ethiopia. The first time we succeeded the synthesis of the nanoparticle in our laboratory by using the medicinal plant. And you see, and after that, it is TEM analysis of zinc oxide nanoparticle. While compared to the when you are seeing the sizes of the nanoparticle, you see zinc oxide nanoparticle size is very small and compared to the silver nanoparticle, because the size variation depends upon their concentration we are using, and concentration of salts we are using, temperature we are maintaining. We are different temperature, like the pH and temperature, all those things which can control the synthesis of size of different sizes of the nanoparticle. Here, while you are seeing zinc oxide nanoparticle, the size of the zinc oxide nanoparticle is 8 to 24 nanometer. Because that's why we are succeeded to synthesize the nanoparticle in our lab. We succeeded in our laboratory by synthesis of nanoscale nanoparticle. You see, after the synthesis of nanoparticle, you see, we are having the 3 samples are there. We are having the 2 nanoparticles are there. One is aqueous extract. Because you see, we should check preliminary before going to anti-inflammatory activity. We should check cytotoxicity of a nanoparticle. Here, important point is we are preparing these 2 nanoparticles by using aqueous extract. That's why we should check cytotoxicity of 3 samples. One is aqueous extract, another one is silver nanoparticle, another one is called zinc oxide nanoparticle in the raw cell lines. Because what is the meaning of raw cell lines means raw cell lines are the model cell lines which used to study inflammation. If you want to study inflammation, the raw cells are a group of macrophages which are responsible for upregulation of inflammation. That's why we are selecting these raw cells for to evaluate the anti-inflammatory activity of nanoparticle. Here you see we are having 3 samples are there, aqueous is there, silver nanoparticle is there, zinc oxide nanoparticles are there. Here we are to treated with we are treated our raw cell lines. Here we have taken 3 different concentration. You see 6.25 to 100 microliters of 100 microliters of sample, aqueous sample. In the same way, this is also we have to take the concentration from 6.25 to 100 microgram per ml of plant as well as the silver nanoparticles are taken to check the cytotoxic effect of nanoparticle. You see, the based upon the result, you see aqueous extract does not showing much cytotoxicity. That's why it is safe. These plant extracts are safe resources for synthesis of the nanoparticle. That's why we succeeded for the synthesis of the nanoparticle for getting the positive result of anti-inflammatory as well as the anti-cancer due to biocompatibility nature. Here, you see, it does not show any cytotoxicity effect against normal cell lines. That's why it does not show any cytotoxicity. That's what is called biocompatibility nature. While you are seeing the silver nanoparticle, the silver nanoparticles, when you concentrate from 25 microgram to 100 microgram, it is showing cytotoxicity up to 80% of cell viability. That's why it is also no problem. And you see, and the zinc oxide nanoparticles also, it is also showing less cytotoxicity. You see, based upon these results, based upon this cytotoxic effect, here our 3 samples are safe to work for to check anti-inflammatory activity. Here, one important thing is the silver up to 100 microgram, no problem. But when we are coming to zinc oxide nanoparticle at 100 microgram per ml, it's showing the cytotoxicity. That's why here for further analysis, we are taking sample 1 to 50 microgram per ml. You see, 1 to 50. This is the cytotoxicity effect of different concentrations of aqueous extract, silver nitrate, as well as the zinc oxide nanoparticle. You see, based upon this slide, we concluded that all 3 samples are furtherly used for to prepare all these 3 samples because TBAQ aqueous extract are the good source for the synthesis of the nanoparticle to evaluate anti-cancer as well as the anti-inflammatory activity. And you see the nanoparticle which are synthesized from the terminal brownie also showing less cytotoxic effect on raw cell. That's why you see the concentration from 6.25 to and 50 microgram per ml. Because you see, while you are showing this this graph, the silver nanoparticle at 100 microgram is showing cytotoxicity. That's why it is better to take to minimize the concentration at 50 microgram per ml. That's why in our experiment we are taking sample range from 6.25 to 50 microgram per ml, because we can go for the standardization. You see, this is the nano cytotoxic about active, because it does not showing any cytotoxic effect against macrophages. When you are showing, you know, you have to observe. You observe clearly. We have ABAQ does not showing any cytotoxic effect on macrophages. Are you getting? You see, this is a very important image because the TBAQ does not showing any cytotoxic effect on macrophages. That's why we furtherly carried out anti-inflammatory activities against this extract. And the same thing, this extract showing safety. That's why this extract also useful for the useful for synthesis of the silver nanoparticle as well as zinc oxide nanoparticle. You see, when you are seeing a result with the silver nanoparticle, this silver nanoparticle at 50 microgram per ml, it does not having much effect. But when we change the concentration from 50 microgram to 100 microgram, it's showing a cytotoxic effect. That's why the silver nanoparticle at 100 microgram per ml is showing much cytotoxicity against RASL. That's why we should exclude 100 microgram per ml for our activity. And same thing, zinc oxide nanoparticle, it does not showing any cytotoxicity effect up to 100 microgram per ml. But to standardize the anti-inflammatory activity of both silver as well as the zinc oxide nanoparticle, we are taking concentrations of 3.25 to 50 microgram per ml to check the anti-inflammatory activity. In the same time, you see, this is the RASL, the RASLs treated with the aqueous extract as well as at 50 microgram per ml. It does not showing. RASL means normal cell, MDB, MV cell means cytotoxic cell. That's why you see our cells, our nanoparticle, it does not showing any cytotoxic effect, but it act on cancer cells. You see, this is that's why our RAS cells does not showing any cytotoxicity, but it's showing cytotoxicity against MDB, MB231 cells. Here MDB, MDA, MB cells means it is a type of breast cancer cell. These cancer cells are responsible for progression of the cancer. That's why our sensitized green as well as the silver nanoparticle, green sensitized zinc as well as the silver nanoparticle showing much cytotoxic effect against MDB, MBA 237 cells. You see, among the tested nanoparticle, zinc oxide nanoparticle showing significance anti-inflammatory activity as well as significant cytotoxic effect compared to remaining thing. You see, this is after confirmation of cytotoxic effect of a nanoparticle, we selected all nanoparticle because all these nanoparticles does not having any cytotoxic effect. But here silver nanoparticle showing cytotoxic effect more above 50 microgram per ml. That's why we are taking concentration from 3.125 to 500 microgram per ml of different concentrations for to check anti-inflammatory activity of a nanoparticle. You see, while we are doing anti-inflammatory activity, first we have to take the RASL. These RASL treated with the LPS. You see, LPS means lipopolysaccharide. This LPS is responsible for to upregulate the inflammation. This is called basal control. This is called LPS control. You see, in our experiment we are having the three treated things are there. One is basal thing means without treatment. Another one is control means treated with the LPS. And another important thing is treated with plant extract or treated with nanoparticle with the LPS. You see, in our important thing is in the presence of LPS, because you see what is the meaning of LPS? LPS means lipopolysaccharide, which is isolated from the gram-negative bacteria responsible for upregulation of inflammatory mediators. You see here when we are seeing this image in basal control means we don't have any nitric oxide. You see in the first slide, this slide explain about effect of this green sensitized nanoparticle on nitric oxide production. You see in figure A, figure B, figure C. Figure A against aqueous extract and the inhibitory effect of aqueous extract against nitric oxide and inhibitory effect of silver nanoparticle against nitric oxide, as well as the inhibitory effect of zinc oxide nanoparticle against nitric oxide production. And based upon these results, we concluded that zinc oxide nanoparticle showing more inhibitory effect on nitric oxide synthesis. It is called, it's showing dose dependent. When you are showing result among all the tested three samples, zinc oxide nanoparticle dose dependently decreasing nitric oxide production. And you see another next slide, when you are going for nitric oxide levels also, you see these nitric oxide levels also decreased with the help of zinc oxide nanoparticle. Okay, this is another important thing. You see, and here important thing is production of PGE2. Okay, you see, already we confirmed that our nanoparticle, it is a safe. We are used for development of anti-inflammatory as well as anti-cancer agent. That's why preliminarily, you see, you know very well, we are having the during chronic inflammation, the COX-2 as well as the LOX is activated. You see, when COX-2 activated, it produces the prostaglandin. That's why the preliminary anti-inflammatory mediators are prostaglandins as well as the leukotrienes. That's why first we have to check the preliminary anti-inflammatory activity of these nanoparticles. When we are checking these nanoparticle with aqueous extract, among all the tested 3 samples, TBAQ, silver nanoparticle, zinc oxide nanoparticle, and the PGE2 exhibit a good inhibitory effect on production of zinc oxide nanoparticles, showing more inhibitory effect on production of PG2. You see, while you are seeing this diagram, we are using different concentrations of a nanoparticle with aqueous extract. While you are checking, you see percentage of inhibition. Here, zinc oxide nanoparticle exhibiting more percentage of inhibition compared to silver nanoparticle and compared to aqueous extract. That's why, based upon our experiment, zinc oxide nanoparticle showing more anti-inflammatory effect when compared to silver as well as the aqueous extract. And you see, based upon the result, we concluded that the zinc oxide nanoparticle exhibiting a dose-dependent nitric oxide activity as well as the PGE2 inhibitory production. You see, because here important point is we are having 3 samples are there, aqueous extract is there, and zinc oxide is there, as well as silver is there. Among the tested 3 samples, zinc oxide nanoparticle exhibiting promising dose-dependent nitric oxide as well as the PGE2 inhibitory effect at 50 microgram with less IC50 value. Okay. Because among the 2 sample, among the 2 here, PGE2 here you see PGE2 production is less when compared to nitric oxide, because the zinc oxide nanoparticle decreasing more amount of the nitric oxide when compared to the PGE2, because this is a result we are getting, because based upon the rubber result, what we are concluding, zinc oxide nanoparticle exhibiting promising dose dependent nitric oxide inhibitory effect. How much the percentage? 64, and PGE2 production 48. That's why we concluded that zinc oxide nanoparticle showing more anti-inflammatory effect when compared to a tested silver as well as the aqueous extract, because it's showing good nitric oxide independent activity at 64. Then you see, based upon this result, we can go for we selected zinc oxide nanoparticle to check cytotoxic effect. Here, zinc oxide nanoparticle showing good cytotoxic effect, 88% of against breast cancer cell when compared to the normal cell line. That's why here, based upon our result, here zinc oxide nanoparticle showing prominent nitric oxide inhibitory activity, PGE2 inhibitory activity. That's why these also act as a cytotoxic effect against MDB, MBN cell. That's why, based upon our result, we concluded that zinc oxide nanoparticle can be used as a potential anti-inflammatory as well as a cytotoxic drug for development of novel drugs for treatment of inflammatory as well as inflammatory related breast cancers. Recommendation, you see, we are furtherly, we've only done in vitro studies only. We are not conducting any in vivo studies. Furtherly, we want to plan to conduct acute as well as the subacute studies, and we have to go for animal models of in vitro anti-inflammatory activity as well as animal models of breast cancer activity. And we should also check desensitized nanoparticle with other biological activities like antibacterial activity and antifungal activities also. That's why you see, your important thing is the green sensitized nanoparticle having more advantage when compared to the other nanoparticles. Thank you very much. The presentation forward. Hello.
Yeah.
Sir,
Proceed, sir.
Yeah, finish, madam.
Okay. Thank you very much, sir, for the
please.
excellent talk.
Yes,
Is
sir.
there any questions? May I ask?
Yeah, please, madam. Please.
Yeah. Is there any questions? Okay, ma'am. Welcome.
Good afternoon, sir.
Tell me, madam. Yes.
Sir, actually, the seminar was, the presentation was very good.
Yes.
It was very interesting.
Okay.
I just want to know that you just the nanoparticle itself has got any cytotoxicity for that, you will find it with the normal, the macrophages.
Yeah, madam, what I done, madam, first of all, before developing of any anti-inflammatory drugs or anti-cancer drugs, first our nanoparticle, it should not have any cytotoxic effect. That's why I synthesized the 2 nanoparticle by using the green synthesizer, and after the synthesis of the nanoparticle, I checked the cytotoxic effect by using the raw cells, madam. Because raw cells also a type of the macrophages which mediate in immunology. Which mediate.
For that, you will be using your sample.
Yes, yes, sir. For that, yes, madam. Yes, madam. I checked the cytotoxic effect by using raw cells.
Okay.
Yes, madam. Because once it is showing, once it does not have any cytotoxicity up to 50 microgram to 100 microgram, we furtherly carried out our work with further mediators, madam, like PGE2 as well as nitric oxide and other cytokines. And it is showing good result. You can furtherly go for to check cytotoxic effect against breast cancer.
Thank you, sir. Thank you.
Okay, ma'am.
Thank you. Okay. Sir, what type of plant you are synthesis, sir?
Madam,
what type of plant? Yeah.
Madam, you see, I am using here, madam, terminia species, madam. Terminia species, madam. Already in India also we are using terminia species to relieve cough as well as inflammatory related diseases as well as like corona also. That's why in Ethiopia traditionally they are using Termina brownie. This Termina brownie they are using for traditional user for treatment of cough as well as some inflammatory diseases. That's why based upon the traditional information, based upon the traditional ALR information, I selected Termina brownie, madam, and I selected this plant and make into the aqueous extract. And this aqueous extract I'm using for the synthesis of zinc oxide as well as the silver nanoparticle, madam. Because these extract it contains the more amount of the secondary metabolite. More amount of the secondary metabolite like flavonoids as well as the tannin. These flavonoids and tannins act as reducing agent as well as the capping agent. Madam, here one important advantage is it does not have any cytotoxicity. It is having more biocompatibility.
This process is a very tedious one.
Yeah. Yes, madam. We can go for standardization, madam. We can go for standardization of plant extract and we can go for standardization of silver also, madam. Because, madam, you know very well, we have to take different concentrations of the plant extract also. We should take different concentrations of different micromolars of silver as well as the zinc also, madam. We can go for the standardization with the different concentrations of plant extract, different molarities of salts, as well different pHs and different temperatures. And after that standardization only, madam, we are getting nanoparticle, madam. But for getting all this standardization, we took 3 to 4 months, madam. Then we succeeded to get nanoparticle.
Sir, excuse me.
What about
which plant pot is used?
Madam?
What about the green synthesis?
Green synthesis means, madam, that's my madam. Madam, what is green synthesis, madam? By using the plant extract, because I selected the Termina brownie based upon the traditional important, and after the selection of this plant, I make into aqueous extract. This aqueous extract, I go for phytochemical analysis, qualitative phytochemical analysis and quantitative phytochemical analysis. Based upon this phytochemical analysis, we find out flavonoids, terpenoids, and polyphenolic compound. These flavonoids and polyphenolic compound act as reducing agent, madam. Act as reducing agent as well as a capping agent. You see, here we are using zinc acetate and silver nitrate. Here, silver nitrate and zinc acetate are bulk molecule. Bulk salts, madam. When these bulk salts adding into aqueous extract while magnetic stirrer, you know very well, magnetic stirrer while boiling, automatically 30 minutes, madam, the preliminary color is changed. Why color is changed means, you see, madam, when we are adding silver nitrate and zinc acetate, it is breakdown into silver as well as zinc. Okay, once it became zinc as well as the silver metals, it became reduced with help of the phytocompound. These phytocompound donate electrons and it became silver nanoparticle. That's why our plant extract, it acts as reducing agent to make formation of silver nanoparticle from the bulk molecule, and it also acts as capping agent, madam. Because you see here, important thing, here we are not using any drug. Yeah, we are not using any drug. Here our plant act as a drug. Here our plant act as a drug, and the plant act as also a reducing agent, madam. That's why due to this reducing agent, it acts as a silver nanoparticle.
Thank you.
Sir, good evening, sir.
Good evening,
Which plant pot is particularly used, or a whole plant?
Madam, bark, madam, because madam, it is based upon the traditional important, madam. It is based upon
So only bark is having so many medical medicinal importance.
Yes, madam. Because what is important thing is here traditionally they are using bark for treatment of inflammatory related diseases as well as fungal infections as well as tumors. That's why here I selected the bark to check the anti-inflammatory activity of nanoparticle.
Sir, you are listing out some of the phytochemicals.
Yeah,
so
yes,
flavonoids, alkaloids, which is actors particularly you are analyzing for FTIR.
Yes,
what is the function? Which phytochemicals can act as a reducing agent or a capping agent?
Very
is there
good,
any?
Yeah, madam. Phenolic compounds, madam. Majorly phenolic compounds are act as a reducing agent as well as the capping agent, madam. Yeah.
So it can donate the electrons to the.
Yes,
okay.
Madam,
so particularly the phenol compound is present in the bark in a higher quantity.
Madam, yes, madam, I'm explaining you a simple thing. You see, already you know very well. First, we are preparing plant extract, madam. Okay, madam. Are you audible, madam?
Yes, sir.
Madam, first we prepare the plant extract. This plant extract, we can go for preliminary phytochemical analysis, and after preliminary phytochemical analysis, we can go for UV as well as the FTIR. And GCMS also. But based upon this information, we got one idea because it containing some secondary metabolite. Okay, madam. These secondary metabolite, more predominant secondary metabolites are flavonoids as well as the polyphenolic compound. Already we are having so many scientific reports. These flavonoids and polyphenolic compound act as anti-inflammatory as well as the anti-cancer as well as the antioxidant agent. That's why based upon our preliminary data, our preliminary analysis as well as based upon our phytochemical analysis data, it contains some polyphenolic compound as well as flavonoid. These polyphenolic compound as well as flavonoid, okay, while it interact with zinc acetate as well as the silver nitrate, okay, it reduces silver into silver nanoparticle and zinc into are you getting, madam? Zinc into
yes, sir, zinc.
Silver into silver nanoparticle. And while this conversion is called bioreduction, madam. It is called phytoreduction and bioreduction. This is confirmed by the color changes.
Okay, sir.
By the color changes, and after the color changes, you confirm that nanoparticles are formed, and after that we can go for a FTR analysis. During FTR analysis, some functional groups are shifting, madam. While group shifting is there, you have to missing some functional groups in while you are doing FTIR for silver nanoparticle as well as zinc oxide nanoparticle, because when we are comparing with aqueous extract as well as the silver nanoparticle as well as zinc oxide nanoparticle, some functional groups are missing. Some functional groups are missing in silver nanoparticle as well as the zinc oxide nanoparticle, because it is interpreted that those missed functional groups are responsible for synthesis of the nanoparticle.
Okay, the missing is responsible for the activity.
Yes, madam, because madam, those missing functional groups are act as reducing agent.
Okay, so that's why that the zinc oxide is having more potent than comparing to the silver.
Yes, madam. Yes, madam. Yes, madam. Yes, madam.
Okay, yes, thank you, sir. Thank you so much.
Yes, madam.
Any questions? So now I request Dr. Malarvili madam, who is the HOD of Biochemistry Department, to respond the speech of Dr. K. Krishna.
Madam, unmute, please unmute.
Good afternoon, everyone. Actually, we are doing the same nanoparticle synthesis in our lab. For even MSc students they are doing.
Okay, madam.
Actually, when we listen to, all MSc students, everyone are listening to your speech, okay. And it was really thought-provoking, and actually we got so many information from your speech. And actually, we have already got some project also in the same thing, like SEM, TEM and all already we have done. But after listening to your speech, it was very good to know that some functional groups can be seen by FTIR, and then the shape, size, everything can be same. All those things we have come to know, and our students and faculty members are really enjoying your speech, and also they got very good information. Thank you so much, sir.
Yes, madam. Yes, madam. Yes, madam.
Thank you, ma'am. Thank you.
Thank you very much. Thank you very much.
Thank you. Thank you, sir. Now I invite
Thank you, madam. Thank you.
Dr. A.V. Srinath, Assistant Professor, Department of Biomedical Engineering, Sri Venkateshwara College of Engineering and Technology, Pondicherry, to present his presentation. Sir, please.
Selina sir.
Oh, yeah.
Selina sir.
He's presenting.
Hello.
Hello, yes, please, sir. Please, sir.
Sir, it is audible to you, sir, madam?
No, audible to us, but your slide is not seen.
Okay. Please wait. Hello.
Sir, we share the PPT, sir.
Sir, Selina sir.
Mute, share pannu.
Unmute பண்ணிக்கோங்க sir.
Please unmute your mobile audio.
Hello.
So we share the PPT in the screen, sir.
Okay.
Oh.
Thank you. Shall I start, ma'am?
You start the press. You start.
First of all, many thanks to the organizers and the coordinators for doing this great job in on recent innovation on biomaterials in current scenario. I myself, A.V. Srinath, Assistant Professor, Department of Biomedical Engineering, Sri Venkateshwara College of Engineering and Technology. My research topic, my paper topic is gap junction connectivity mechanism in human Purkinje cells, a cell pair model to define cardiac memory. Next slide, please. Yes, thank you. So, the introduction. It is estimated that by 2025, cardiovascular disease would account for 37% of all fatalities in the industrialized world, resulting in 2.5 crore cases per year and becoming the leading cause of death. In India, the annual number of deaths from cardiovascular disease is projected to rise from 32.6 lakhs in 2010 to 67.7 lakhs in 2025. Okay, cardiac memory refers to the T-wave inversions that result when normal ventricular activation resumes following a period of abnormal ventricular activation. Abnormal ventricular activation is most commonly induced by ventricular pacing, but can also occur in the setting of transient left bundle branch blocks, ventricular tachycardia, and also intermittent ventricular pre-excitation. Recognition of pacing-induced T wave inversion is the diagnostic tool to identify and control the cardiovascular problems. Next slide, please. Okay, my problem definition. Suppose if somebody is affected by cardiac problem, immediately the first tool to diagnose is an ECG signal. The ECG signal is nothing but a rhythmic representation of cardiac function or electrical representation of rhythmic activity of cardiac function. So once the cardiac regular rhythm gets disturbed, it is reflected in the ECG. It is self-differentiated to identify any cardiac abnormality, and it was diagnosed by the physician. For example, a patient admitted for a cardiac problem, generally the doctor used to say that after seeing the ECG pattern, he may have some cardiac arrest either yesterday or day before yesterday, maybe within 15 hours or last night, something like that. So that indicates the remembrance behavior of the heart. That is called the cardiac memory. What happened in the past. Our aim of the research proposal is to identify cardiac rhythmic and also the cardiac memory behavior of the heart. So based on this research, this topic is framed. Next slide. Okay, what is cardiac memory? Actually, a modified T wave, that is you are seeing in the slide, there is a red arrow is showing that the T wave is inverted. A modified T wave on electrocardiogram and vector cardiogram is identified as a cardiac memory. In the first diagram, you see the normal normal ECG pattern. In the second and the third, you see the abnormal ECG pattern. That is called arrhythmias. Is generally ECG as arrhythmias. So that is seen even sinus rhythm resumes after irregular myocardial activation. Specifically, the sine rhythm of the T wave tracks the QRS vector of the irregular impulse. That is, with the repeated simulation of ventricular activation or pacing, there is a variation in T wave magnitude. T wave inverted and retained, even the external pacing was removed completely. This retention of inverted T wave is remembered or memorized for several days. Say for example, up to 21 days are shown here. Next slide, please. What is gap junction channels? As you know, gap junctions are membrane channels that mediate the cell-to-cell movement of ions and small metabolites in the heart. Gap junctions play a crucial role in impulse conduction. Studies over the last decade have revealed that gap junctions are encoded by a multi-gene family known as connexins. There are at least 15 connexin genes in the vertebrate genome. In this regard, gap junctions are analogous to synapse, like neurons. Synapse transmissions, like in the brain, on the other hand, are mediated by the neurotransmitters. Wide gap junction conductance accomplished by direct flow of ions among the cells are synchronizing. Okay, oscillations of a wide variety of cell types, gap junctions allows vast regions of heart tissue to contract as a single unit, a functioning syncytium. Next slide, please. So these are all my literature survey. Next slide. Next slide. Next slide. Okay. So this is my project workflow. First of all, modeling of human Purkinje. Actually, my project is only around the Purkinje cells. That is not about the SA and atrial and ventricle cells. Only about the Purkinje cells. First of all, the modeling of human Purkinje cell are defined from existing latest models. Then the next step is to derive the single cell response for simulation from MATLAB software and verify the results with the existing recent models. This process is very important to carry out all other remaining analysis successfully. And after the study of gap junctions in the cardiac cells, the third step is the coupling of cells through the gap junction conductance to define the proper synchronization between the coupled cell pairs, that is combining two cells in series manner. The analysis was done by the same cell pair coupling, that is Purkinje and another Purkinje cells are connected and the analysis can be done. The next, the fourth step is by introducing the external pacing. That is, we earlier discussed that is external pacing was used for this. With Hebbian adaptation, when without Hebbian adaptation was studied. Hebbian is a process. He is the first person to determine there is a memory process or done like brain. There is a memory process also there in the heart. It is a first and foremost analysis done by the Hebbian adaptation. Finally, the cardiac memory was proved from the result. Next slide, please. Next slide, please. Is it audible? Next slide, please. Thank you. So, all our results will be focused on electrophysiological behavior of a single cell. That is, Cm of dV by dt is equal to I ion plus I stim, where Cm is a membrane capacitance and V is the membrane potential and T is the time. I ion is the sum of transmembrane ionic currents, and I stim is the externally applied stimulus current. Next slide, please. Next slide. Thank you. And if the two adjacent cells are coupled, then the dynamics will be like this. That is, C1 of dV1 by dt is equal to I1 of ion kinetic plus Gj. Gj is representing the gap junction conductance. V2 minus V1. That is the cells. Say for example, cell A and cell B are connected. And then the potential difference between the two cells are calculated. And in C2 dV2 by dt, that is I2 ion kin plus gap junctions. Now V1 minus V2 are calculated. Like that, the series of cells are connected and calculated. Then, next slide. Okay. The cardiac system comprises, that is the comprises with SA node, right and left atrium, AV node, Purkinje fibers, and left ventricle, etc. Each side runs with its own intrinsic frequency. The intrinsic ranges of each cell's beats in a different frequency, but it is wonder to see that the whole heart beating in a common frequency of 72 BPM. That is, we know. That is referred to as a normal heartbeat. As seen in the figure, the variation in the intrinsic frequency and shapes are shown, but it is well reflected with a single uniform frequency, and that is electrocardiac, that is ECG. Next slide, please. Okay, here we are seeing the single cell response for Purkinje cell, and it was modeled from the Stewart et al. 2009. And this is a single cell with 35 BPM. That is 38 BPM means beats per minute. 35 beats per minute. This is the response for single cell response. Then, next slide. Okay. So it is understood from the electrophysiology of the cardiac system that the cardiac cells are communicating each other. Like neuron cells, the cardiac cells are also communicating each other through a specific protein structure called gap junction channels. The different regions of the heart contains specific connection expressions, and gap junction conductance ranges in Siemens as mentioned in the table. Actually, we are only concentrating on Purkinje cells, that is connection expression of CX40 and CX43 and CX45. The conductance ranges between 30 picosiemens to 200 picosiemens. Okay, next please. So this is the electrical representation of the two coupled Purkinje cells. The first one is the Purkinje cell of cell 1, and second one is the Purkinje cell of cell 2, and these two cells are connected with the gap junction conductance. It is represented in the variable resistance. Now we have single cell response for the Purkinje cells. Our primary aim is to couple the cells which beats in a common frequency. Next slide, please. Okay, here you are clearly seeing that here we connected a pair of cells with 48 BPM and 42 BPM. That is two cells. One cell is running with 48 BPM Another cell is running with 42 BPM. So actually these models are done not in a real-time manner. The models are get from the website source, and the codings are calculated with the Euler's method. By MATLAB software only, we will get these type of results. So cell 1 with 48 BPM and cell 2 with 42 BPM with the gap junction conductance of 140 picosiemens, and they are running in a different frequency. As you can see in the figure, they are running in different frequency. They are not running the same phase. So they are not in a common phase shift, and the phase difference of the responses are expanded continuously. You see responded continuously seen in the figure. That means the cells are connected by the gap junction, but these cells are not properly coupled. This condition leads to irregular heart rhythm. Okay. Right. First, we select the gap junction conductance value as 140 picosiemens from the given range. Next slide, please. Sorry. No. Existing style. Existing. Slide number 18. Please come to slide number 18. Okay. Thank you. Okay. Now here, then we started to adjust the gap junctions values to 90 picosiemens. When cell 1 with 48 BPM and cell 2 with 52 BPM, after seconds, as you see in the figure, after 5th second, these two cells are synchronized in a frequency of 50 BPM. This is the first and foremost analysis we have done. Already we have certain range of gap junctions values for each cells. By using that range of value itself in the synchronization takes only for some type of pair of cells, not for all. Okay, so we tried a number of pair of cells with the different BPM and gap junction values to identify a proper coupled pair of cells. The synchronization happens, that is coupling is happen in a particular range of gap junctions only. In the previous slide, go to the previous slide, please. Previous slide, 17. Okay. No, 17. Slide number 17. Okay. First, we select the gap junctions. As you see in the Purkinje cell data, first we select the gap junction values as 140 picosiemens from the given range. Now, the two cells are connected in the different frequencies. The action potential propagates within the cells, but in a different frequency. It is not necessary for us. Come to the next slide, please. See, the primary purpose of the study is to select the gap junction conductance value that beats in a common frequency, as shown here. Here, the gap junction conductance value is set to 90 picosiemens, as you see in the diagram. 90 is picosiemens. Now, the cells beats in a common frequency of 50 BPM. If the gap junction conductance value is set to 95 picosiemens, then the cells oscillates in 51 BPM of common frequency. From this, we have identified that even though we have nominal range of gap junction conductance value, the coupling of cells happen only in a certain range of gap junction conductance. That is, before adjusting the gap junction conductance ranges from 80 picosiemens to 90 picosiemens, and after adjusting the gap junction conductance ranges from 78 picosiemens to 105 picosiemens. So the main purpose of the study is applying the pacing or disturbance in the normally conducting two pair of signals. Only after the two connected cells attains a common frequency pattern, so that it is easy to study the disturbance effect. So we are constantly made several simulations with 10 lakh iterations for each pair of cells. Okay, to complete a single simulation, it takes around 10 to 12 hours. Please, next slide, please. Okay, that is Hebbian adaptation on cardiac cells with the external pacing. Till now, the coupling between the cells are defined. Now, to prove the memory property of the external pacing was introduced in the normally coupled pattern by Hebbian adaptation method. Till now, we are connecting two Purkinje cells in a series manner and try to run in the same frequency. Now, the next step is to giving an external pacing is given to the normally connected cell, and we will see the analysis part. Next slide, please. Okay. So our main objective is to prove the memory property in the heart by Hebbian learning method. Hebbian learning is a process of updating the weight between neurons. As you know, in neurons, in the neural network, for each training samples to strengthen the connection between the neurons. It is denoted by delta WAB is proportional to V of t and VB of t. WAB is the synaptic efficacy. VA of t and V of t are the presynaptic and the postsynaptic activity of the cells. The cells in cardiac system are also same as that of the neuron cells. Here, the gap junction connections between the cells were tuned to provide proper propagation of activation pulse between the cardiac cells. When the impulse between the two neurons strengthened, the repetition or memory behavior happens in it. Likewise, the propagation process strengthened between the cardiac cells, repetition of same activation pulses generated. For example, when we are in a great loss, our brain drastically disturbed by thinking again and again about the loss. But within certain period of time or days, we will come back to normal stage. Likewise, our heart signal pattern also disturbed by these losses, and there is a disturbance in the activation pattern of the pulses. This can be come back to normal within hours or days, or even permanently abnormal. That is called heart attack. By Hebbian learning, the original pattern will be remembered quickly. That is the foremost work of my project. The next one. Next slide, please. First of all, the 4 stages of the mechanism is explained here. In the first stage, the results obtained from the stage 1 with synchronized common frequency are taken. In the second stage, the external pacing was introduced for a short and long period. Then the input pacing is removed. In the third stage, the disturbed sequence by pacing are identified and adaptation process starts. Finally, the cells beat in common frequency again by with and without Hebbian learning are analyzed. Please, next slide. Okay, this is a clear pictorial representation of my analysis. Here, the Purkinje cells are beats with 50 BPM and 60 BPM. As you can see in the figure, that these two responses are not in a common frequency. The first condition is without Hebbian learning. Here, in the first stage, the first 2 pulses shows the synchronized form of the cells. See, in the first 2 pulses, I think you are seeing the slide. The first 2 pulses are in a synchronized manner. Then in the second stage, that is external pacing stage. The second stage, the external pacing of 0.5 milliamps of short input. That is any loss, any problem, any external disturbances occurred to our heart, like that. 0.5 milliamps of short input was introduced for 1 second in the 1.6 seconds of the pattern, and removed at 2.6 seconds. That is for 1 second it is retained in the stage 2. See, as you see in the third stage, disturbance in the pattern. See, in the first stage, the 2, that is 50 BPM and the 60 BPM pulses are in the same frequency, and in the second stage, the external pacing was introduced, that is indicated by an arrow mark, and it is removed again by another arrow mark. And in the third stage, the disturbance done, it happened up to 8th second. You see, from 3.7 to 8 seconds, these 2 signals are not in a common frequency. In the fourth stage, the pattern again follows the common frequency. Again, it tries to follow the common frequency. Here, the pattern resumes after 8 seconds without Hebb learning with the common frequency of 54 BPM. That is, without Hebbian learning, it tries to come back normal and after 8th second. Okay, next slide, please. In this slide, next slide. Okay, in this slide, that is slide number 23. Okay, here the Purkinje cells are beats with 50 BPM and 60 cm, respectively in those same frequency, right? In the second stage, again, the external pacing of the same 0.5 milliamps are introduced for 1 second, that is with 1.6 seconds on a pattern, and removed at 2.6 seconds. Like in the third stage, the disturbance done by the external pacing was shown. Here, In the fourth stage, as you see in the slide, the waveform is come back to normal within 5th second. In the previous slide, it is come to normal at the 8th second, but here it come back to normal at early at 5th second itself. Because using the Hebbian learning concept, we are reducing the time. That is, we are reducing our emotion. We can come back to normal normally. The same thing is happened here. Here the pattern resumes after 5th second itself with Hebb learning with common frequency of 54 BPM. That is 3 seconds more quicker than without Hebbian learning. Next slide, please. Again, this is for a long input method. The same analysis was done for long duration input pacing, and the results are shown here. Like the first condition is without Hebbian learning. Here in the first stage, the first 2 pulses shows the synchronized form of the cells, as seen in the, as we see in the last slide. Then in the second stage, the external pacing of 0.5 milliamps of long input was introduced for 4 seconds. That is, in the previous slide, we give the input external pacing for 1 second. Here we are going to give the external input for 4 seconds. That is 1.8 seconds of the pattern and remove at 5.8 seconds. In the third stage, it shows the disturbance done by the external pacing from 5.8 seconds to 1.8 seconds. Sorry, 18.16 seconds. In the fourth stage, the pattern again follows the common frequency. It is without a Hebbian learning method. Next slide, please. Next slide. Okay, that is slide number 25. The second condition is with Hebbian learning. That is, we are seeing the same analysis for short input and long input. First, we seen about short input. Now we are analyzing about long input. That is the thing. Here, the second condition is with Hebbian learning. Here, in the first stage, the first 2 pulses shows the synchronized form of the cells. Here, the same method is carried out. Here, come to the disturbance in the pattern stage. Here, it shows the disturbance done by the external pacing from 5.8 to 15.9 seconds. Okay, in the fourth stage, the pattern again follows the common frequency. Here, the pattern resumes after 15.9 seconds with Hebbian learning in common frequency of 54 BPM. Thus, 2.7 seconds reduced with the Hebbian learning, which indicates the original pattern resumes or remembered more quickly when compared to without Hebbian learning. From this analysis, we are concluding that memory property was proved in Purkinje fiber cell of cardiac system by Hebbian learning method. Next slide. Discussion. So far we modeled the cells for Purkinje cells, and we find the role of gap junction on cardiac communication was studied. And then the Hebbian adaptation on pair of cells, the same cells were implemented and tested, and the cardiac memory property was proved for the pair of Purkinje cells. And we are doing our project in the remaining cells, that is SA node and the atrial cells and ventricle cells. Finally, we will do the same process for the remaining cells also. Thank you. Thank you for your patience. Any queries, please ask.
Thank you, sir. Thank you.
Thank you,
for a
ma'am.
wonderful presentation.
Thank you, ma'am. Thank you.
It is only in the prototype type. Is it? Are you trying in the market side?
No, ma'am. It is only in the prototype. It is only in the simulation method.
Yeah.
Yes,
You won't test for the human?
No, ma'am. It is not given to us because it is. We only give the idea to the medical person.
Oh, I see.
Yes, ma'am. Yes, ma'am. Yes, ma'am.
If you are having any plan to go to the medical side?
Yes, ma'am. We have plan. We are applied for the patent for this method.
Oh,
After, if it is processed, then we will give the medication with this method, madam,
okay,
to reduce the heart attack problems.
Congrats for you getting the patent.
Thank you, ma'am. Thank you, and thank you for the opportunity, and thanks to Dr. Muthulingam sir for giving this wonderful opportunity. He is one of my oldest friend. Now we are also in a good friendship manner. Thank you, sir, Muthulingam sir.
Thank you. It's a nice work, sir. You may go into the medication side.
Yes, ma'am. Yes, ma'am. Thank you. Thank you.
Thank you all the participants for your presence and make this day a grand success. Tomorrow you will have some more instructions. The feedback form is sent to you through the link tomorrow. Tomorrow you have an important section in the first section. The Patent Right Office is going to give some awareness program. It is up to 10 to 11:30 and we have wonderful lectures there. So don't miss it. The attendance is very important for giving the certificate, and the registered candidates, the registered participants may get the certificate. So register yourself through the link. It is very important. So you got two more certificates, one from Indian government of India, and another one is Government of Tamil Nadu. So you are very lucky. The patent office is going to give one more certificate to all the participants. So it is very useful for you. It's very important. Thank you for your kind cooperation. Thank you very much. We will see tomorrow with a new hope and a new enjoyment. Thank you very much. Thank you.
Thank you, ma'am. Thank you.
Thank you.